Dear colleagues and dear participants, I think we can start this event. It is time to start. Good afternoon and good morning, according to where you are. It is my pleasure on behalf of the organizing committee to welcome you to this online final event of the XFACTORS H2020 project, which is jointly organized with the third European Conference on Xylella fastidiosa. If I can share the screen. And this conference followed the previous that we held in Mallorca in 2017 and the one in Ajaccio in 2019. Unfortunately, due to the COVID emergency, we had to adapt to an online format of the conference, but we still hope that you can engage a fruitful discussion with the speakers and with the attendees. And it is for this reason that we decided to use Twitter and the platform Zenodo for sharing the poster session in order to promote visibility and public discussion on the topic presented. 1 second, if I can share. Okay. So this third conference on Xylella fastidiosa is promoted by the European Food Safety Agency in collaboration with the XFACTORS and with all several other research initiatives that you see here below in the slide that cooperated for the organization. The first 3 days of this event will be dedicated to XFACTORS. So today, Tuesday and Wednesday afternoon, we will present the results gathered in these 4 years H2020 project. Then the event will continue on Thursday afternoon and Friday all day with the European conference. Before spending some words to illustrate the consortium XFACTORS, I want to thank and express my gratitude to EFSA for its spirit of cooperation and for being willing to organize jointly this event, and in particular to the EFSA staff for taking care and supporting the major part of the organization of this event. I want also to thank all the representatives of the research initiative mentioned represented in this slide. So all the other projects, you will hear about some of these projects during the next days. And then I want to thank the reviewers who volunteered to revise and to evaluate all the contributions we received for this conference. And of course, I want to also thank the chair of the session. I believe that being an online format, chairing the session will be even tougher than the normal conference because we have to keep time, the session in time, and an active discussion. I will go now into a couple of slides to explain and to present for those that are not directly involved in the XFACTOR consortium, the mission of the consortium and some details on this consortium. Briefly, I will not go into the details of the work plan and on the activities that we have done in XFACTORS because there will be plenty of speech during these 3 days that about the scientific objectives and the tasks we developed during the project. But I will just focus on few general information. The project, the consortium include almost 30 partners from all over the world, as you see here in this slide, from European countries, but also we had partners from California, Costa Rica, Brazil, and Taiwan. Most of the partners include research institutes operating in different experts in different disciplines, from microbiology to entomology, expert in modeling, in epidemiology, and as well as in communication. We were also happy to have on board two international organizations, the Joint Research Center and the European Plant Protection Organization, as well as some private companies or association of growers. More specifically, some companies working on the diagnostics or association of nurseries or farmers working in plant health and certification of plant propagation material. The mission of the project has been to develop new knowledge on the different aspects characterizing the disease caused by Xylella fastidiosa. So we tackle the pathogen, the bacterium, the vectors, and the host plants, and also the environment that contribute to the disease development. The main objective was then to use this knowledge for implementing control strategies, which is still a major challenging in the management of Xylella fastidiosa disease. We, as a project, and as I will show in this slide, one of the key features of XFACTORS consortium has been that we have established a well working research network in Europe. Almost all the major centers working on the topic were included in this network, and this made a very fruitful collaborations. For sure we exploited the collaboration with the previous H2020 project tackling Xylella fastidiosa, the project Ponte. Our project was complementary to this to Ponte, and the collaboration was we benefited from the achievement obtained from Ponte, but also we continued the experiment of Ponte. As you may know, several experiments dealing with Xylella fastidiosa and with perennial crops need multi-year experiment that could not be concluded in the framework of Ponte. So these have been continued with XFACTORS. Then during the implementation of the project, we have a very intense collaboration with different kind of initiative, both for Marie Curie, for example, fellowship, or MSCA RISE project like CureXF. In this project, had the objective to transfer knowledge to third countries, and even if in the last period the secondment activities have been slowed down, in the first part of this project we had a very fruitful collaboration with people coming in the XFACTORS partners institution to learn what we were doing for Xylella fastidiosa in Europe. We had also a collaboration interaction with the EFSA network, mainly for the diagnostics. In this case, the contribution was more for developing validation data for the diagnostics, but also to contribute to the capacity buildings of the personnel involved in the laboratories of the member states by organizing several international training. There has been also a continuous collaboration with EFSA, and regarding this, my colleague Antonio Vicente will present more in details our contribution and collaboration on Wednesday, and then we have several points of contact with the Cost Action Europe Fund dealing with Santa Manadace, And so even if these bacteria have a different behavior, they are the same family of Xylella fastidiosa, so we had useful interaction with this cost action as well. And as I was mentioning, there was one of the Marie Curie fellow that was developed in strict collaboration with the XFACTORS regarding the genetics of Xylella fastidiosa. In that, as you will see from the presentation, in these days, several contributions are the results of a co-funding by national and local initiatives. So XFACTORS has boosted several activities and has collaborated with several activities also at the national and the local level. And for the future, the achievement and the partners of XFACTORS have in different ways cooperated for the new the ongoing projects on Xylella fastidiosa, namely BioVexo or the EFC multi-X. You will hear about these two projects in the next days during the conference. This collaboration was extremely important to avoid duplication in the research program, in the new research program, as well as to develop and exploit the knowledge that was already achieved in the framework of XFACTORS. Then the other aspect I want to highlight about the consortium is the active knowledge transfer we have put in place during the project. Since the beginning, periodically we have met with the DG Agri and DG Santé and in Brussels.
Dia, vai a chiudere la porta del terrazzo? Chiudi la porta del terrazzo.
Excuse me, there are some interference. Please mute your microphone. So, sorry, I was saying that one of the important activity of knowledge transfer was during the policy meetings we had at the end of each reporting period, in which the researcher involved in the project presented the latest information to the policy officer, and this was not only at the European level but also at the national level. Several scientists have been interacting with our phytosanitary authorities, especially in those countries dealing with outbreak of Xylella fastidiosa, in order to ensure a fast transfer of the knowledge into then for the implementation of the policy and for the implementation of the measure for the eradication and containment of Xylella fastidiosa. Then I will say that in some cases, some of us has dedicated more time to the dissemination than to the research. And I will take this opportunity to thank all the colleagues that, besides doing the research, spend a lot of time to support the training, meetings, and webinars and seminars to inform stakeholders, farmers, growers, and the citizens. So this was also very active in the project in different countries. So with this, I conclude my welcoming, and I would give the floor now to Gisela Quaglia. She's a research officer at the DG Agri. The DG Agri is the part of the office of the Commission that funded XFACTORS, and as I said before, they are following the outcome of the project during the whole project lifetime. So I want to thank Gisela for being available to give her welcoming this afternoon, but also for being available on Friday to report about the new Horizon Europe program. So she will give insights on the new research program. So I stop sharing my screen and thank you, Gisela. The floor is yours.
So good afternoon. It's with great pleasure to be here today for the final meeting of the project. So on behalf of the European Commission, I want to thank the project coordinator, Maria Sabonari, for the invitation. I will try to share my presentation. So my name is Gisela Quaglia, and I work as Research Program Officer at the Research and Innovation Unit in DG Agri. So our unit is responsible for the development and implementation of European research policy in the field of sustainable agriculture and forestry. I imagine that we all will prefer to have this meeting physically, but despite this context, we have ahead a very comprehensive agenda and program that looks very promising. So I will be brief to let the time to researchers and project members to show the results obtained after more than 4 years of projects. But let's take a moment to go back in time and recap since the first detection of Xylella in Puglia in October 2013. Almost 7 years ago. At the beginning, we count with Ponte, as Maria just mentioned, which a third of a project was dedicated to Xylella and was funded by Horizon 2020 at that time. But in 2015, we decided to open a call for proposals specific for Xylella that was open under Horizon 2020. The project should use multi-actor approach, which requires a solid collaboration between research, plant health authorities, and farming sector, and also international collaboration was encouraged at that time. This consortium, XFACTORS, that was the winning proposal. It's count with 29 partners and 14 countries. That makes the project quite ambitious. But moving up to the present times, I also want to highlight, as Maria just mentioned, that we can see that additional efforts were made during these years. New projects related to Xylella were funded under Horizon 2020, and as Maria mentioned, there was a great interaction among these projects, and this is a remarkable thing for this kind of actions that avoid overlapping and creates and transfer the knowledge from one project to the other. At the end of the timeline, you can see the logo of Horizon Europe. That is our new research framework, research and innovation framework program, which will bring further funding opportunities. I will give more details about this on Friday afternoon. So if you stay with us till the end of this conference, you can hear a little bit about Horizon Europe. So the goals that we set to the project, to XFACTORS, were quite ambitious: to develop knowledge, to fill research gaps, to extend knowledge on biology, on genetics, on the pathogen, on the vector, on the host plant, but also to provide tools for surveillance programs for early detection, to integrate this research data with models to develop best risk assessment. But an important thing that also was mentioned by Maria is to establish a platform for adequate communication and dissemination, and establish also this research network that it was so needed for Xylella. I will not give I will not go in detail with the results, the interventions and the presentations foreseen in the program will provide us a great overview of the results. But I will like just to mention that you can see the link on the bottom of the screen that we just published in Cordis, that is the European Commission Public Repository, an article showcasing the results and achievement of this project for the general audience. And we want to thank also the project for organizing the policy session last year for Commission services. I can say that the scientific developments from this project have contributed largely to science-based policy. When I was preparing this presentation, I found the slides presented by my colleague, my previous colleague in the position, Eleonora, for the kickoff meeting that was in 2016, I think in Bari. And she includes this quote from Henry Ford that I think is still applicable for this project and for today. Coming together is a beginning, keep it together is progress, and working together is success. Before finishing, I would like to take this opportunity to thank my colleague, George Perdue, the project officer from the Research Executive Agency, who has followed the project since the beginning, and also colleagues from DG Santé and also DG Agri, who had interacted with these projects quite fruitfully. I'm very much looking forward to hear the presentations today, and I wish you all a very fruitful meeting. Thank you.
Thank you very much, Gisela. Thank you for attending and being interested. It is important for us to have the DG Agri and the European Commission to listen to our results. And I take this opportunity to thank also Eleonora Ganci, who followed the project in the past, as well as Pasquale Di Rubo from DG Santé that now moved to other DG. So thank you. And I will now give the floor to Giuseppe Stancanelli from the Alpha unit of EFSA. This unit had a major role, as you will see in the next day, for the update of the scientific opinion on Xylella fastidiosa, the host plant database. And Giuseppe in particular led the organization of this conference, so my deep thank you for him for taking all the efforts to organize this conference. Giuseppe, the floor is yours.
Thank you very much, Maria, for giving me the floor. So the first things I would like to ask all the speakers to mute their microphones so we don't hear all these noises when another one is speaking. That's for the whole conference. We have to pay attention to this. So why we're here? We are here because we're organizing this conference with XFACTORS. And we are particularly happy to welcome you to this joint event, which will go on for the whole week. So the first 3 days will be about XFACTORS, final results. This is a very important project for us that really support European risk assessment on Xylella, decision-making, the fight of this bacterium in the last year, together with the previous results of the Ponte project. And then we will continue with the Xylella conference, the 3rd European Xylella conference, with around 900 registered participants, not all in the same time, because everybody will attend different sessions. And the participants are around 50% from research and academic, but then a lot of participants also from private sector, NGO, national protection organization, international organization, farmer, nurserymen, risk assessor, risk manager. So really a composite participation from 66 different countries. And so we really hope this will provide a platform to present and discuss results of research and also to discuss new ideas. So welcome everybody and enjoy this meeting that I hope is very fruitful for the control of this very important plant pathogenic bacterium. Thank you, Maria.
Thank you, Giuseppe. And since we are already with some minutes of delay, can I share the screen? I will just briefly summarize the program before to start the first session. So please, can I share my screen to show the program in brief? I'm sorry for the... I asked to Davide to give me the right to share the screen. Okay, I will start even without the presentations, just, okay. So just to summarize this long event, we are starting today with a session on biology and genetics of Xylella fastidiosa. Tomorrow we will continue with the results gathered in the work package of the project dealing with the vectors and with the related control strategies. And then in the late part of the afternoon, a session regarding the control of Xylella in the host plant. So all the attempts and the experiments that have been done. And then we will complete our final meeting with the fourth session on Wednesday regarding the implementation and harmonization of diagnostic protocol, surveillance and modeling. And we will also conclude with two speeches by François Peter and Antonio Vicente, remarking the contribution of the project to the National Plant Protection Organization and to the European policy. In the last day, we will also have a contribution regarding the communication. Communication in science is now a hot topic also for the COVID-19 emergence. So we will hear about some suggestion and the overview regarding the communication and the issue related to the communication in science. Then the second part on Thursday, the second part of the conference of the event will start with starting the European conference. We will have on Thursday two sessions. One on advances on pathogen biology, ecology and genetics, and then understanding vectors biology and pathogen transmission. Both sessions will be in the afternoon. Whereas on Friday we have all-day event. We will start in the morning with a session on epidemiological modeling, surveillance and early detection, and then continue in the afternoon with a scientific session on sustainable management strategies, which will be then followed by a stakeholder session in which there will be a discussion among different stakeholders and the scientists. So and this session that will be then concluded, as we said today, with Gisela Quaglia, who will present the Horizon Europe program, and the colleagues from DG Santé also reporting the overview from the policy part. It is important also to mention about the poster session of this conference. All the posters you can find at this hashtag Xylella21 posters. It is important that the authors can reply to the original account of Plant EFSA tweet. In this way, you can contribute to the competition that has been launched with the online session of the poster. So the posters are all on Twitter, and then from Twitter you can get to the link to the DOI and the link to Zenodo, where you can see both the abstract and the slide of the poster, which in this case are in format of a PowerPoint in slides. As I said, EFSA launched this social competition. A reward will be given to the authors of the most interesting and the best presented work. This reward will be based on Twitter analytics that will be downloaded on Thursday. Sorry, can you close them? Can you mute the microphone, because there are some disturbance? And as I was saying, the Twitter analytics will be downloaded on Thursday, 29th of April, and then the winner will be announced in the morning of Friday. The winner of the competition will have an interview with EFSA that will be then published on EFSA, made available on the YouTube channel of EFSA. Another important part for this conference is the interactions with the speakers. All the attendees have a Q&A box, and you can make a question to the speakers through the Q&A, to this Q&A box. Here the instruction: you have to type your question, select all panelists from the windows, and then send your question. It is important to say that we expect a large numbers of questions to be sent through this system, and so only few will be answered orally during the conference, whereas all the remaining questions will be replied after the event. The organization committee will organize the answers along with the proceeding of this conference, which are now online. So you can also, going into the website of EFSA or XFACTORS, you find also the proceedings of this conference. And as stated here, this event is about research results, challenges, needs, and priorities. So all questions that are outside of these topics may not be replied. And I think we can start now with the first session. Will be chaired by Donato Boccia, who is from the XFACTOR coordination team. He is the responsible of our institute, CNR Institute in Bari, and Maria Agnes from INRA. I thank both of them for being available for chairing this session. In particular, Maria Agnes, who is also the work package leader, and so had invested lots of efforts in coordinating all of the activity on biology and genetics in the project. I give you I'll stop sharing my screen and I give you the floor. I think the first to start is Donato. Donato, please, the floor is yours.
Yes. Thank you, Maria, for this. Thank you, Maria, for this introduction. As you said, I will share the chairmanship of this session with Maria Agnes Jack, but we agreed that I'm the one supposed to start with the first, by chairing the first presentation. And I will go straight to give the microphone to the first speaker, since we are already, as usual, a few minutes later, according to the program. So the first speaker of this session that I remember is achieving practical outcomes from genetic and biological studies. So the first speaker is Anne Sicard. Anne Sicard, she has a double affiliation, the University of California, Berkeley, where I think she did the first part of the work that she's going to present, and the Plant Health Institute of Montpellier, where she is currently working, if I'm not wrong. So Anne, please, you have the floor, and all the speakers will have 15 minutes of time for the presentation, and 2 minutes before the end of the time, I will advise them of the time. Okay, thank you, and please, Anne, go ahead.
Good afternoon, and thank you very much, Donato, for the introduction. Can you hear me correctly? Yeah, okay.
Yes, go ahead. Okay.
Thank you. So today I'm going to present you a project that I've worked on during my postdoc in Rodrigo Almeida's lab, together with our collaborators in Italy. So this project deals with the introduction and adaptation of Xylella fastidiosa to olive trees in Apulia. So as all of you know, Xylella fastidiosa has a very broad host range as a species, having been described as being able to infect 595 plant species belonging to 85 plant families, and causing severe damage in some of its hosts. However, the host range of a given strain is much more limited, especially in terms of disease. In other words, one given strain causes disease in a few plant species. So for instance, among the host plants represented here, only Polygala myrtifolia, Myrtus communis, and olive trees are among the hosts of the Italian strain Bedono. So the broad host range of Xylella fastidiosa as a species is indeed linked to its great genetic diversity. So here you can see the genetic diversity of Xylella fastidiosa based on the analysis of 72 strains coming from several plant species. So the three main subspecies classically described in the literature, which are Fastidiosa, multiplex, and Pauca, can be distinguished, along with a few unresolved taxa. So if we look at the pangenome of all these strains, we can see that the core and soft core genome, which corresponds to the genes present in all the isolates analyzed, include a bit over 900 genes, which approximately corresponds to 40% of the genome. So while the shell genome, which corresponds to genes present in a subset of the strains, include over 3,200 genes, and the cloud genome, which here gathers the genes present in less than 10 of the strains, include over 7,200 genes. So just to be clear, the genes present in the cloud and in the shell genome corresponds to genes present in one or in a few isolates, which explains their large number. So basically, that means that the majority of the genes are present in only a subset of the strains analyzed, highlighting the high genetic diversity of Xylella fastidiosa, which allows its adaptability. So major concern regarding this pathogen progressively expanding its geographical range worldwide, and notably within the Mediterranean, as depicted here, is its ability to form novel associations with host plants. So indeed, as it progresses into these naive environments, Xylella fastidiosa may adapt to new host plant species. However, the genetic mechanisms driving Xylella fastidiosa adaptation to novel plants or environments remain unknown. So to tackle this important question, we have chosen to study the recent outbreak of Xylella fastidiosa in Apulia. So on this map, you can see the progression of the infected areas in Apulia, and more precisely, the positive foci detected since 2013 in this region. So between 2013 and 2017, 75 infected olive trees were collected within the affected region. So most of the samples were first cultured before being sent for sequencing, while 10 total DNA samples were sequenced directly. So you can see here the geographic distribution of the collected samples. So each spot corresponding to one isolate, and the colors corresponding to the sampling time point. So previous studies have shown that the dono, which is the reference isolate in Apulia, belong to the subspecies Pauca. So I've therefore represented here a phylogenetic tree regrouping all the Pauca isolates for which a complete genome was available. So I've used as an outgroup a strain belonging to subspecies Fastidiosa. So as you can see, our Italian isolates, which are in green, cluster together, which confirms that the epidemic in Apulia is indeed linked with the introduction of a single Pauca genotype. We can also see that the Apulian isolates are very close genetically to 3 isolates from Costa Rica, which here appear in blue. Indeed, previous studies based on MLST analysis have shown that all these isolates belong to the same sequence type named ST53, which means that they have the same allelic profile for 7 housekeeping genes. So if we look at the close-up here, we see that the Costa Rican coffee strain, COF0407, is the closest strain from the Apulian isolates. So let's now have a look at the ST53 pangenome. So we see that the core and subcore genomes comprise 2,190 genes, which corresponds to nearly 88% of the bacterial genome, with each genome including a bit less than 2,400 genes. So that means that the majority of the genes are present in all or in almost all isolates. And on this Venn diagram, you can see the repartition of the genes within the ST53 isolates. So the close relatedness between the Italian isolates and the Costa Rican coffee strains pushed us to further compare these isolates. So the idea was to see if we could find some mutations that were shared by all Italian isolates in comparison to the Costa Rican strains. So for that, we used 2 different approaches. So here you can see the results of the first approach, which is based on a core genome alignment. So each purple line here corresponds to a SNP, that is a mutation affecting a single nucleotide. So the reference genome being the Costa Rican coffee strain. So we can notice that most SNPs are present in only 1 or in a few isolates. But some of them are present in all Italian isolates and absent in the Costa Rican ones. So these mutations are quite interesting since they are shared by all the isolates infecting olive trees in Italy and absent in the Costa Rican isolates. So they could be involved in the adaptation of Xylella to olive trees in Apulia. So we then use a pipeline to compare the Italian isolate with the genome infecting coffee in Costa Rica. So this pipeline was developed for the analysis of microbial resequence samples that are only slightly divergent from the reference sequence. So it presents several advantages since it allows us to determine not only SNPs, but also deletions. Moreover, we obtain a complete annotation of the results. So as you can see on this table, we know which genes are affected, and in the case of SNPs, if the mutations are synonymous or non-synonymous. So I've summarized here all the mutations common to all the Italian isolates, so still in comparison to the Costa Rican coffee strain. So as this table is a bit hard to read, we have represented here all these mutations. So we have found 54 plasmidic genes that were deleted in the Italian isolates. These genes are primarily involved in plasmid maintenance, conjugation, and replication of the plasmid. We also found 63 genes in the chromosome that evidence mutations in all Apulian isolates when compared to the Costa Rican ones. So these mutations correspond either to indels or to SNPs. So here you have the functions of the chromosomic mutated genes. So as you can see, a fair amount of these genes encode for proteins of unknown function that are either complete or incomplete, and a few of them are also phage-related. Apart from those, the mutated genes were involved in metabolism, DNA replication, signaling, gene expression, cell wall growth, and pathogenesis. So as positive selection is considered being the primary mechanisms of adaptation, we next looked at the genes under positive selective pressure in the Apulian datasets. So as expected, we found that most of the genes were under neutral selection, while 84 genes were found to be under negative selection, and 115 genes were found to be under positive selection. So we found a few functional categories that showed at least a 25% enrichment among the genes under positive selection in comparison to the genes under neutral evolution. So I've indicated here the enriched categories with cell motility displaying the highest enrichment. So what's really exciting here is that among the genes found to be under positive selective pressure, 6 of them were also found to present shared non-synonymous mutations among the Italian isolates in comparison to the Costa Rican ones, making them good candidates for Xylella adaptation to this new environment. So what are these genes? So 2 of these genes correspond to Xylella two-component quorum sensing system, RTFC and RPFG, the global transcriptional regulator, CRP, an ATP-dependent helicase, an ABC transporter substrate binding protein, and an enzyme involved in the biosynthesis of bacterial cell wall peptidoglycan. So finally, we use these isolates to date the introduction of Xylella fastidiosa in Italy. So to do so, we first checked for the presence of temporal signal before using a tip dating approach. So this approach allowed us to date the introduction of Xylella fastidiosa in Apulia around 1994. So to conclude, we have first confirmed using whole genome sequencing of 75 isolates that the outbreak in Apulia is indeed the result of a single introduction of a Pauca genotype from Costa Rica. The close proximity of the coffee-infecting strain from Costa Rica, together with the repeated detections of Xylella-infected coffee plants in Europe, point towards an importation of a Xylella-infected coffee plant, likely as a non-mental plant at the origin of this outbreak. So we estimated this introduction to have occurred around 1994. This introduction was then followed by subsequent establishment and adaptation to this new environment, as suggested by the evidence of genes under positive selection. So finally, by using a combination of population genomic approaches and evolutionary genomics methods, we identified a short list of genes that could be involved in the adaptation of Xylella to this new environment. So the next step would be to confirm their role in the adaptation of Xylella to olive trees by reverse genetics. With that, I would like to thank all the persons involved in this project, and in particular Rodrigo, Mathieu, and Andreina, as well as our collaborators in Italy, Donato, Pasquale, Maria, Annalisa, and Giuliana, and my peer in France, Claire. So this project was funded by the H2020 programs through the XFACTORS project and through a Marie Sklodowska-Curie fellowship. And finally, I would like to thank the committee meeting for giving me the opportunity to present this project. And thank you for your attention.
Hello, everyone, and thank you, Donato, for this quick introduction. And today I'm going to present you our last results on the introduction and the emergence of Xylella fastidiosa subspecies multiplex in France and its probable dates and scenario of introduction. First, let's look at the world distribution of Xylella. You can see on this map, represented by circles, the fossils detected before 2013, and by stars, the one detected after 2013, which is the date of the first detection of the bacterium in the European environment in Italy. Sorry? As you can see, As you can see, Xylella is widely spread in the Americas and the south of Europe. Since this bacterium is widely disseminated by humans, first works are allowed to infer the introduction routes of the different subspecies from one country to another, and to estimate the date of introduction. And these works all converge in one direction. The different subspecies of Xylella would all originate from one of the American continents, and they would have then been introduced from South or Central America to North America or vice versa, and later on to Europe and Asia. Now, if we focus on the European situation, you can see on this map the different subspecies represented by different colors, as well as the subgroups of second type detected in each region. Among the outbreaks, the subspecies multiplex, here in red, has been detected in Portugal, mainland Spain, Balearic Islands, Picard region of France, Corsica, and Italy. And 4 different second types have been identified. The same can be seen for the subspecies Pauca in purple, which was detected in 4 regions of Europe, and for which 2 second types have been identified. So, the detection in the environment of different second types belonging to the same subspecies, and some of which are dispersed in several countries or regions, suggests that multiple introductions have occurred in Europe. And some of these introductions have started to be estimated. A first study dated the introduction of the subspecies multiplex in Corsica in 1985. However, it should be noted that the estimation of this introduction was made on the only basis of the number of plants detected infected, without taking into account genetic data, such as the second type of the strain. So, in line of this first work, Howa consisted in studying the diversity of the subspecies multiplex in France in order to date and estimate the most probable invasive scenario. You can see on this map of the Provence-Alpes-Côte d'Azur region on the left and the Corsica region on the right, the distribution of samples detected as infected by Xylella between 2015 and 2018, as part of the French official monitoring plan of Xylella. In France, the vast majority of samples, more than 95%, were contaminated by the subspecies multiplex, blue dots on the map. And out of a total of more than 28,000 of samples analyzed, 917 samples could be fully typed, and 2 second types were identified, which are the ST6 and ST7. And both second types were already known in the USA, where ST6 was detected on almond trees and ST7 on a wider variety of hosts, like almond trees, but also olive trees and red oak trees. Moreover, out of the 970 samples detected as positive in France, 52 strains were isolated, and this from 50 different plant species. So in order to estimate the introduction in France of each ST6 and ST7 strain, we sequenced all of the 52 strains we had at our disposal. To date, the divergence between strains, which is a lower estimate of the date of introduction. The French genomes, as well as all the publicly available ones, allowed us to build a dataset of 82 genomic sequences of the subspecies multiplex, and isolated from different countries, USA, Brazil, Spain, Italy, and of course France, between 1983 and 2018. Based on this data, a core genome on 1.68 megabases was defined, from which 13,818 SNPs mutants were extracted. Based on this SNP, a tip dating analysis was performed. And using BEAST software and knowing the date of isolation of strains, this analysis allowed to calculate the substitution rate of genomes and then to estimate the date at which the strains have diverged from a common ancestor. The substitution rate of the subspecies multiplex was calculated to be 3.22 10⁻⁷ substitution by seed Bayer, which is close to the one previously calculated in other studies for the subspecies Pauca and Fastidiosa. Then, the divergence depth between our French strains and their closest relatives Here, American strain was estimated at 1,971 for ST7 and 1,987 for ST6. However, the use of genomic data alone on only 52 strains did not give us enough information to trace back the dissemination routes of the subspecies multiplex in France. Thus, in order to trace back the dissemination routes of Xylella in France, we needed to use other data and more accurate typing tools. Moreover, as it was not possible to try again to isolate Xylella from samples that were frozen for a long time, we developed a new MLBA scheme, which is based on the typing of rapidly evolving markers and to implement it directly on plant samples kept in a freezer. MLBA, for multi-locus VNTR analysis, is a method that consists in searching and amplifying repeated motifs, or microsatellites, whose number of repeats varies from one strain to another, showing a recent evolution. A VNTR profile is then defined for each strain by concatenating the number of repeats of each VNTR, and then the profile obtained allows us to analyze the diversity present in our dataset. The MLBA scheme developed here consists of 13 VNTRs dispersed on Xylella genomes and including 10 VNTRs previously described in the literature for other subspecies, but also present in the subspecies multiplex, as well as 3 new VNTRs specifically designed for the multiplex subspecies. This scheme was first tested in silico on genomes and in vitro on strains to validate that in addition to be present, the VNTRs were polymorphic. Then, the possibility of using this scheme to do direct typing was tested in 2 steps. First, by analyzing spiked polygala matter weight. This allowed us to validate that the presence of plant material did not cause any major inhibition of the reaction. Then, we typed Xylella in 13 naturally infected blood samples for which a strain was isolated in the place. And in these 13 cases, the MLBA profiles obtained for the strains and the infected plant were strictly identical, and this allowed us to validate the reliability and robustness of our method. Now, if we go back to the French situation, as I mentioned earlier, we had 970 samples collected between 2015 and 2018. And we successfully typed nearly half of these samples with our MLBA scheme, which was really good, taking into account that the sample was stored in the freezer during several years and that all the 13 VNTR PCRs needed to be successful for a complete typing. Thanks to these results, presented here in the form of a minimum spanning tree, we can observe the evolutionary links between the samples. On these trees, one circle represents an haplotype, and the size of the circle is proportional to the number of individuals sharing these haplotypes. So, compared to MLST, which only identified and differentiated ST6 and ST7 samples, the MLBA identifies 320 haplotypes, allowing a more detailed analysis of the diversity of Xylella in France. Now, if we look at the year of the isolation of the strain, we can see that the founding haplotype of ST6 was identified during several years, as well as the ST7 one. Also, we found haplotypes at the end of evolutionary lineage that had been sampled as early as 2015. And this suggests that we are sampling in an already diverse population and that we are not observing an epidemic that is spreading. So, in order to identify the point of introduction of the subspecies multiplex in France and then to trace back the dissemination of Xylella, an ABC analysis was performed on the MLBA data. This type of analysis consists in evaluating the most probable scenario among a pre-established list. And for this purpose, the ST6 and ST7 individuals were each divided into 3 groups, and these groups were obtained using the results of a DAPC analysis. The objective of this subgrouping was to try to understand the direction of the spread of the subspecies multiplex between PACA and Corsica regions. Then, the most probable invasion dissemination scenario was searched using the American strains ST6 and ST7 at our disposal as ancestral population. We use these strains as ancestral population because multiplex subspecies would have originated in the USA and would have then been introduced in Europe, and because among the strains we had at our disposal, the tip dating analysis that I presented to you previously validated that these strains were the closest relative to our strains. Even if the MLBA data obtained for each of the STs were analyzed separately, the same scenario was identified as the most likely for ST6 and ST7 individuals. This scenario will be composed of a first introduction of a population we named P2 in PACA. From this P2 population would have diverged according to a bridge ahead scenario, a second group we named C1P1 that we found in Corsica and PACA. From the P2 population, a third group we name C2 would have diverged and moved in Corsica. To conclude, I will summarize the most probable scenario of introduction and dissemination of the subspecies multiplex ST6 in France. First, we will consider that the bacterium was introduced from the USA, directly or not, and this could have happened from 1987, maybe from Polygala. Why do we make this assumption? Simply because before the detection of ST6 in France, this second type have only been identified in the USA, and that our tip dating analysis showed that among the analyzed strains, the closer relative to French strains were American. Moreover, because a recent study also suspected the Southeast American area as the origin of the multiplex species present in Europe, based on phylogenetic cross-construction. And it is known that genetic improvement work was carried out on Polygala in Florida before it was widely commercialized in Europe. And Florida is a region where ST6 is not only present, but also considered as endemic. We can thus imagine a first introduction in PACA from there. On this bridgehead population, a group would have then diverged and established both in PACA and Corsica, mainly around Ajaccio. And finally, a third group would have later diverged from PACA to spread in Corsica, and this time mainly around Porto Vecchio. Here, the elements that could confirm this hypothesis of the direction of the dissemination of the subspecies multiplex were brought by Chauvin et al., which reported a trade of plant material only entering in Corsica, and this mainly from the continent. In addition, in older study from Jean Monod and Nathalie concerning the introduction of plant in Corsica, they highlight the massive introduction in the 70s to 90s of alien plant species, among which Polygala could be part. This scenario is also coherent with introduction toward port areas such as Ajaccio and Porto Vecchio. The invasion scenario of ST7 strain is essentially identical to the ST6 one, except that the initial introduction may have occurred earlier, and that the dissemination of individuals of each group being more dispersed for this second step, we can imagine that the trade of plants initially took place on a larger scale. For now, the subspecies multiplex was mainly found in PACA and Corsica area of France, but it was modeled that the entire French climate could be favorable for its spread and survival. So the next step should be to anticipate and consider that the bacterium could be a threat for other crops and the environment.
1 minute.
Sampling effort should be maintained and expanded. So a new strategy should be set. For example, the establishment of sentinel plants may be an option to follow the spread. And to finish, I would like to thank you all the collaborators I've been working with for this project from INRAE, ANSES, CIRAD, and CIRM-CFBP. And thank you all for your attention.
So the first question is, how confident are you of the year of introduction of Xylella in Puglia?
So we ran 5 independent runs to get the states, but as I've shown, we have a confidence interval which ranges from 1950 to 2007. Sorry. So there is some uncertainty about the date for sure.
Okay, thank you. And I think that we can also make this question also to Enora, if you like to add some comments.
Yes, we also run several tests, and the confidence we have for ST7, it is, the diversion date will be 1,971 for ST7, but it ranges from 1,924 to 1,993. And for ST6, we date the divergence at 1,987, and we are confident between 1,974 to 1,994. So it's a lot of numbers.
Another question, thank you for your answer. Another question for Anne. Anne, what was the method that you used to detect positive, negative selection?
So we used 2 different statistical methods to look at selection. One was a frequency-based method, which is called Tajima-D test. And we use also a gene-based method.
Okay, thank you. There is still one question for you, Anne. How can we explain high Xylella fastidiosa genetic diversity? It is linked to the age of the bacterium, where age maybe is number of years of the bacterium.
Yeah, that's an interesting question. I think Cindy Maurice and Benoît Mourir have estimated Xylella to be 700 million years old, if I'm not wrong. So it might be actually pretty old, which has led a lot of time for Xylella to adapt to different environments. Regarding the main evolutionary forces that maybe at the origin of this great genetic diversity, we know that recombination plays a major role in Xylella evolution. Notably, we know that it is a natural competence. And also we can think that the positive selection that may act on the different strains infecting like new environments may drive this important diversity.
Thank you. Thank you, Anne. There is another question for Enora. Enora, that's for you. Why so much undetermined in Corsica?
Okay, so to detect Xylella in samples, it's a 2-steps protocol. First, we try to detect the bacteria Xylella fastidiosa using a qPCR from Harper and al. And then we try to type the bacteria to determine the sequence type using a MLST scheme. And so for the first step, when we try to detect the bacteria using Harper qPCR, if the CQ value is higher to 35, it's a kind of a limit of a threshold in which we say that in which we can't say it's a positive samples, but we can't say it's a negative samples too. So we name it undetermined. And it means that maybe it was an error in the manipulation, or maybe the bacterial load was too low to be correctly detected?
Extreme temperature differentially affects growth and survival of Xylella fastidiosa strains. Miguel is from Córdoba, from the Instituto de Agricultura Sostenibile of the Consejo Superior de Investigaciones Científicas. So please, Miguel, please go ahead.
Okay, thank you, Donato, for your presentation. And can you see the presentation?
Yes, go ahead.
Okay, thank you. Good afternoon, everyone, and thank you to the committee organizing for having me here. In this talk, I'm going to present the result we have so far about the response of Xylella fastidiosa strains to extreme temperature. Xylella fastidiosa is a worldwide distributed bacteria. According to the Copenhagen climate map, which Xylella is represented with green points, is present in different climate zones, from oceanic, Mediterranean, tropical, and even semi-arid. And it seems also to be adapted to areas with mild winters. In addition, the consecutive detections of the bacteria in Europe, even new sequence type, reveals the need to the development of risk model for Xylella fastidiosa in relation to environmental factors. These models for climatic suitability showed that most of the European territory may be potentially suitable for the Xylella fastidiosa establishment. And in these models, temperature was one of the main predictors. Also, temperature is one of the environmental factors who strongly affect the wilt disease, since the relation in pathogen and host plants. On one hand, the effect of the temperature on growth and survival were addressed by Fell and Purcell on 6 strains from United States. And the results showed that an optimum temperature for these strains between 25 and 52 degrees in vitro, and that temperatures below 10 degrees and above 54 degrees were limited the growth of Xylella in potted grapevines. And on the other hand, since Xylella is confined in the xylem vessels, it could be also relevant to ask how what is the xylem, or how the xylem temperature differs from the external conditions. And that could be especially relevant for woody crops like olive, almond, or grapes. There are some studies that show that temperature measured in plant tissues do not necessarily correspond with the age temperature. So in this context, the objectives of this study were to determine the effects of the extreme temperatures on in vitro growth and biofilm formation for Xylella fastidiosa strains of the subspecies and sequence type prevalence in the European outbreaks, and describe the dynamics of temperature in xylem vessels and soil root system of an olive tree ocar and determine its relationship with air temperature. To address the first objective, we have chosen 53 strains belonging to 5 subspecies and 15 sequence types from a wide geographical origin and host plants. We have evaluated growth and biofilm formation. At the beginning of the experiment, all the strains were incubated during 7 days in a range of temperature from 4 to 40 degrees. And after 7 days, we measure total growth with the optical density at 600 nanometers and quantification the biofilm by the crystal violet protocol. And at this point, in order to see the effect of the extreme temperatures on Xylella fastidiosa cell survival, the planktonic cells were transferred into a plates, and those plates were incubated for another 7 days at the favorable temperature of 28 degrees. In this slide, we present the effect of the temperature on total growth. As an example of 18 strains of the total of 33 of this study and belonging to the 3 main species. In general, we can observe how all the strains we have used had the optimum temperature in the range of 20 and 30 degrees. And there are some difference between strains, but also we can observe a certain pattern between subspecies, as we will see later. In addition, we have observed that temperatures of 10 degrees and 36 degrees impaired or limited the growth of some of the strains of Xylella, and temperatures of 4, 6, and 40 degrees and impair or inhibit the growth of Xylella fastidiosa strains. If we look at the biofilm information, and compare with the result obtained in growth, we can see the same results. Those strains incubated at temperatures below 10 degrees and above 56 degrees were impaired to form biofilm or have limited this ability. So after assessing the effect of the temperature in the total growth and biofilm formation, we want to determine if the absence of growth at these extreme temperatures were due to a bacteriostatic effect, or this is just because that cells were dead. So we take those microplates and incubated it in a favorable temperature. So in this figure, we present the results of growth. This is the total growth of survival cells after 7 days at 28 degrees. As we can see, those strains incubated at the temperatures below 10 degrees were able to recover that ability of growth again. And this occurs in the 3 main subspecies. So temperatures below 10 degrees do not limit bacterial viability. In contrast, those strains incubated at the temperature of 56 and 40 degrees were impaired to recover that capacity to grow. So temperatures above 52 degrees affect bacterial viability. These results were confirmed also when plating the cell suspension in PD2 and BCA plates. As we can see in these photos, the strains that grew or incubated at the temperature from 4 to 50 degrees were able to form the colonies, while the strains incubated at 56 and 40, it didn't. Regarding biofilm formation, we can observe the same pattern. So that strains that were incubated at temperatures below 10 degrees were able to recover that capacity to form biofilm again. And the strains incubated at 56 and 40 degrees were impaired to recover that ability. If we take a look to the photos, in the first row, there weren't any biofilm formation at the extreme temperature, low or high. And in the second row, we can see when we put those microplates at a favorable temperature of 28 degrees, we can see the typical violet coloration in the strains that were incubated at 4, 6, and 10 degrees. So the conclusions of the first part of the talk were that extreme temperatures, low or high, differentially affect growth and survival of Xylella fastidiosa strains. Bacterial cells incubated at 4, 6, and 10 degrees showed as lower growth rate and reduced biofilm formation. However, at a favorable temperature, these cells were able to recover growth and form biofilm. and an incubation temperature of 36 and 40 degrees for a 7-day period result lethal for most of the bacterial cells. This is the result we have obtained of the response of Xylella fastidiosa to extreme temperature. So our next objective was to determine the dynamics of temperature in Chilean vessels. And since the relation between both factors could be relevant in the disease development of this pathogen. To address the second objective, we have chosen 4 trees from olive oak placed in Córdoba in the south of Spain. And as we can see in this figure, 6 thermocouples were installed per tree, 2 in the branches, 2 in the trunk at 1 centimeter depth, another 2 at 4 centimeters depth, and another 2 per acre, 20 centimeters depth in the soil rot system. And temperature were measured at 10 minutes intervals during 2.5 years. And our interest was to study what happened when extreme temperatures occurs. We have a calculated maximum and minimum temperature per hour, day, month, and sensor. And we have performed an ANOVA analysis, a 24-hour monthly profile of temperature, and a regression analysis to study the relation between the temperature measured in shillings and air. I'm going to begin with minimum temperature, which occurred from November to April. And as we can see in the 24-hour profile, Chilean temperatures were coupled to the air, especially at nights. However, we can see that temperature in branches was slightly lower than in trunk. And the regression models showed that air and Chilean temperature were linearly related. So that means that Chilean temperature measured in branch and trunk progress at the same rate than air temperature. For maximum temperature, which occurred from April to September, we have observed that we can see in this figure that Chilean branches temperature was similar to the air. However, Chilean trunk temperature was lower than the air, with decreases from 4 to 8 degrees lower than the air measured in trunk. The regression models show it for branches similar results that found for minimum temperatures, with a rate of increases close to 1. But the temperature measured in Chilean trunk progress at a lower rate than air temperature. Also, we have found differences between seasons, being the slope of the fitted models lowest in summer, as we can see here, but also for spring and winter we have observed a slope lower. Finally, we want also to study the maximum and minimum temperature in the soil rot system. That's their importance because Xylella is able to move to the roads and overwinter there. So as we can see in this both figure, we don't find no large oscillations.
So Miguel, 2 minutes left, please.
Okay, thank you. Okay, thank you. No large oscillations for maximum and minimum temperatures, and the soil and the sunlight soil sensor the temperature recorded was higher. We have observed a buffer effect in the soil, and we have measured as the soil minimum temperature was higher than the air with increases of 7 degrees, and the soil maximum temperature was lower than the air with decreases of 10 degrees. And we have also observed that the relation among temperatures of soil rot sensor and air was lower in winter and summer. The conclusion of the second part of the talk were that branches temperature was coupled to the air for both maximum and minimum temperature. And for maximum temperature, especially summer and spring, shielding trunk at 1 centimeter depth and the soil rot system and 20 centimeters depth showed a lower temperature compared to the air. We think that this result would allow to the development of regionalized epidemiological models to predict the risks associated with the establishment and spread of Xylella fastidiosa strains in Europe. And thank you to my group and the collaboration, and thank you for your attention.
Caroline Baruch. Caroline, she will give a presentation about investigation of the metabolic network of Xylella fastidiosa responsible of its fastidious growth. Caroline, she's from the Université de Toulouse at INRAE-CNRS from Castanet-Tolosan. Please, Caroline.
Thank you. Do you see my screen well?
Yes, yes, go ahead.
Okay. So good afternoon, everyone. So today I will present the results of the work done in the context of XFACTORS. So we investigated the metabolic network of Xylella fastidiosa and found links to its fastidious growth. I cannot switch. I'm sorry, I can't switch my slides. I don't know why.
Click on the presentation and then.
Sorry?
Click on the presentation and you can use
okay. So, thanks, sorry. So, yes, so high proliferation of, high growth rate is a way to overpass the host immune system, the host immune response, sorry. For example, against immunity molecules such as reactive oxygen species. So it's a bit paradoxical that some pathogens have a fastidious growth. So we qualify fastidious growth as a growth that yields a low maximal population levels and that has growth rates under 0.01 per hour. And so you can have two types of pathogens, those that growing fastly, such as E. coli or Ralstonia solanacearum, which is a plant pathogen, and those that grow very slowly, such as Xylella fastidiosa, but it's also true from human pathogens such as Legionella pneumophila or Bordetella pertussis. And one question we had, is it this low growth, is it because the pathogen requires specific nutritional and environmental specific nutritional and environmental requirements, or is it an interesting property of the pathogen? To answer this question, we use a systems biology approach. So the idea is to use mathematical model to better understand plant pathogens growth and virulence. So we start from the genome, the pathogen, and the host, and we try to predict the phenotype, which is growth and pathogenicity. And for that, we use the metabolic level of the cell and mathematical models. So a classical workflow to perform this approach is first, you have to get the sequence of the sequence genome of your pathogen. Once you have the genome, you have a reconstruction step that allows you to have the metabolic network of your pathogen. Metabolic network can be viewed as a map of all the chemical reactions that can occur inside the cell. Once you have the metabolic network, you transform it into a mathematical problem. And unfortunately, the problem is too big to solve, especially because we are lacking a lot of intracellular data. So we have a simplification step of these models, which allows to perform simulations at the cell level, and we can get from this simulation the fluxes of matter inside the cell. And from these fluxes of matter inside the cell, we can perform several tests in silico that I will develop later. And it helps to gain a better understanding of metabolic behavior of your organism. And this allows to go back to experiments to check the predictions and to better understand the metabolic behavior of the pathogen. And these experiments allows to refine the metabolic network and the simulations. So first, the reconstruction step of Xylella fastidiosa metabolic network. So we use a French strain, which is called CFBP 8418, and it was from Corsica, this strain, and it's a subspecies multiplex. For the reconstruction method, we use an in-house method that relies on high-quality metabolic ontologies on high-quality metabolic networks. So here is the list, and it's mainly from E. coli and Ralstonia solanacearum. And from this, we get a draft metabolic network of Xylella fastidiosa, which contains around 1,000 reactions. But this network is not complete. Indeed, a draft model has many false positives, gaps in pathways, and errors in notations, which gives a non-functional, non-predictive network. And to get a functional and predictive network, we have to perform a curation step that my PhD student Leo did. It took him 5 months, and he manually created the model to check. He checked the reaction accuracy by using databases, experiments, and literature. He used he gap-filled the pathways that were missing reactions, and also he added species-specific reactions that could not be propagated from the models, such as E. coli, and this implies reaction, for example, for fastidian gum or O antigen genes, etc. And so then we got a functional network. And from this functional network, we then did metabolic modeling. For that, once we got a model, we analyzed this model and we look into why Xylella is fastidious. So we check 3 things: auxotrophies, robustness, and efficiency. So first, auxotrophies. So for this, we use graph analysis, and we were surprised, but Xylella is not auxotroph. It has a complete network, but minimal. And the only auxotrophy we could detect was one in the glycolysis or glyconeogenesis, which is the enzyme fructose 1,6 bisphosphatase that was missing. This enzyme is necessary to synthesize exos such as glucose that are necessary for peptidoglycan synthesis, for example, from sugars which have 2 or 3 carbons, such as pyruvate, for example. It's an enzyme that is conserved in all living organisms, and it's the same enzyme whether for animals, plants, or bacteria or archaea. So we were very surprised that we did not find this enzyme, and this enzyme is lost in all Xylella strains sequenced so far. So what's the implication of missing fructose 1,6 bisphosphatase? It's this enzyme is essential to generate exos. This was demonstrated experimentally on Saccharomyces cerevisiae and Clostridium glutamicum. And so we thought, okay, Xylella just needs an exos such as glucose or fructose to grow, and it's exotroph to exos. But then our partner in Angers performed biolog phenotypic microarrays. It's growth assays on 190 carbon sources, and these results, Xylella could grow on amino acids or organic acids, which are not exos. So we concluded that indeed Xylella has no exos auxotrophy. It can have a fructose bisphosphatase activity without this enzyme. So we hypothesize that this enzyme, this activity is performed via the side activity of another enzyme. So the side activity of an enzyme is different from its main activity and is not optimal. And we we simulated in silico what could be the consequence on growth if this FBP activity is performed by another enzyme. And what we could show is that if we divide by 3 the optimal flux matter of our simulations, we can get a fastidious growth that is similar to what is observed experimentally on Fastidiosa strains. So it's a 50-hour doubling time. If we have a normal fructose biphosphatase activity, we have a 2-hour doubling time, which is similar to organisms that are growing normally. So the other 2 things we investigated is robustness and efficiency. So relating to robustness, what we observe is that Xylella has restrictive trophic capacities. It was also confirmed with the biolog phenotypic microarrays. And the reactions that were, for example, present in E. coli and Ralstonia, but missing from Xylella networks, were mainly linked to assimilation of alternative carbon sources like lipids, carbohydrates, and small organic molecules. Also, what we see is that Xylella fastidiosa has many reactions that are only catabolized by 1 enzyme. There is no isoenzyme for this reaction. So more than 90% of metabolic reactions of Xylella are only catalyzed by 1 enzyme, which is not the same in, for example, fast-growing organisms such as E. coli or Ralstonia solanacearum. We saw that this enzyme distribution was similar in Bordutella pertussis, which is a human pathogen that is also slow-growing. We also perform gene essentiality in silico, and what we saw is that Xylella fastidiosa has really not a robust metabolism because on minimal media glucose, 54% of the genes are essential in Xylella instead of 15% in E. coli or Ralstonia solanacearum. And we also looked at fluxes variability, which in a way measures how many parallel pathways exist in the metabolic network. And in Xylella fastidiosa, the fluxes are not flexible compared to Ralstonia and E. coli, which have much more flexibility in its network. So from this, we concluded that Xylella metabolism was not robust, but the lack of robustness does not explain directly fastidial growth. So we took a deeper look into this lack of robustness. And what we discovered is that the enzymes that were missing are the enzymes that are efficient for growth. For example, cobalamin is a vitamin, and the same metabolic reaction can be performed by cobalamin-dependent and cobalamin-independent enzymes for some enzymes. And the cobalamin-dependent enzymes reactions are more efficient. And in Xylella fastidiosa, Xylella cannot synthesize cobalamin and does not possess any cobalamin-dependent enzymes. We got the same results from the cofactor molybdopterin, and globally there's absence of efficient enzymes, cofactors, ubiquinons, etc. So this could also explain the fastidious growth of Xylella. Finally, on the efficiency point of view, we looked at how Xylella is efficient for synthesizes biomass, excrete virulence proteins, and EPS. And we compared it to another pathogen, Ralstonia solanacearum. And what we saw is that for EPS synthesis, Xylella fastidiosa is not efficient because when you have 100 carbons, such as vitamin, for example, 42% of these carbons are lost as CO2 instead of only 28 for Ralstonia solanacearum, which could also explain the inefficiency of Xylella growth and the low OD that can give at the end of the growth curve. And this is in line with the what we can observe. There's a PTRI mutant that has reduced biofilm and protein secretions, and it has an enhanced growth rate and a higher maximal optimal density compared to the wild-type strain. So our analysis is in line with this mutant behavior. So the take-home message is that Xylella fastidiosa lacks robustness in its metabolic network. Either it has restricted trophic capacities, lack of functional redundancy, and a limited flux variability. Also, the absence of key enzymes and cofactors ensuring rapid growth there is an absence of key enzymes ensuring rapid growth. And finally, there is a structural inefficiency that gives a poor yield for EPS synthesis. And thank you for your attention.
So, Miguel, one question is, low temperature viability goes against the idea of cold curing. How low do you think the temperature may need to go to significantly reduce the viability, if at all?
Yeah, I'm not sure about that. We are trying to do and to think how to avoid that, because when you freeze the cells, it's complicated to evaluate that freezing and everything. But we have to take in account this is in vitro site. So with a fresh media, and we also have a wide range of Xylella strains. I think we have to think into or research in order to see if this behavior that we have seen in these strains in vitro could be repeated also in plants. I think is.
Okay, thank you. There is another question. Of course, because the impact of temperature raised a lot of practical interest. So, can you explain the distribution of Xylella in Mediterranean countries is related to temperature, because in the South Mediterranean temperature can reach levels more than 36, which can be lethal for Xylella?
Yeah. We'll see. In our experiment, we have incubated all the strain during 7 days at this extreme temperature. So in a normal Mediterranean climate, where when reaches that 36 or 40 degrees, they are during some hours. This is extreme temperature is punctual, and we have incubated this in this extreme temperature during 7 days. Besides, the second part of the talk could be help us to understand that, because we have seen that in the temperature measuring in trunk was lower than measuring the air.
Yes, I think that the buffer of the trunk is very important. So there is still another, this is a more technical question. Why did you not use exposure window period of 4, 8, and 12 hours for 36 degrees and 40 instead of 7 days? Which are those expected in areas affected by Xylella fastidiosa? I don't know if
yes. Yes. We are working on that. We are working on that because trying to be more realistic in the real world. We are working in more experiments, trying to see how many hours or how many days can be good for the bacteria or can be lethal for the bacteria. We are working on that.
Okay, so there is another question for you, but before I want to give a question to Caroline. Caroline, did you consider in the analysis the small size of the Xylella genome in relation to other bacteria for comparison, I suppose that should be used bacteria with a similar genome size, but with a different growth pattern. What do you think?
So we did not consider the difference of size because we wanted to see where the reduction was of Xylella genome was in the metabolic network. And for the growth rate, it's true that we could compare to bacteria that has a similar metabolic network size. Unfortunately, there is none so far existing in literature, so we could not perform this. And we compared to other pathogens that were slow growing, such as Bordetella, which has also very reduced metabolic network and genome. And we find similar losses, but it's also a slow growing bacteria. So it's probably why.
Okay, thank you. Now we go back to Miguel. Considering the results you just presented, what kind of available climatic data, as air temperature, soil temperature, land surface temperature, etc. What kind of available climatic data would you consider, among other factors, to improve the prediction of Xylella emergence in new locations?
So I think the best way is try to incorporate all the information that we have. The presence of Xylella that we know now and their associated climatic conditions, but also this information that we are generating about the relation of the air temperature with this correction of the air temperature, and also the behavior we have found of the behavior we have found of the effect of the temperature in all the Xylella fastidiosa, trying to considering all the diversity in order to do a more accurate model.
Okay, thank you. So it seems that so far there are no more questions. So I want to just make a comment to the results of Caroline. So if I well understand the metabolic network of Xylella fastidiosa is not optimal for the growth of one bacteria. So I was wondering what could happen having an optimal network, metabolic network, considering the impact that Xylella is able to bring to the plants. I don't know if you have some comment about it.
Do you mean what could happen on its growth rate or on its epidemic status, like how it's going to spread?
Well, on both.
Well, so what we show in our work is that it's not one. For example, we could think that if we add the fructose bisphosphatase, Ambergris bisphosphatase, it can grow much faster, and then it would be easier in the lab to study, but it could have drastic impacts in agriculture. It's difficult because, for example, so what we show in the work is that it's multiple reactions that were excluded from the network, and the sum of all the losses probably makes it fastidious, not only one reaction. But if we add all these efficient reactions, Xylella might grow better, and we will try to see if in mutants we can achieve this phenotype. On the epidemic level, I think there's a trade-off between how fast you kill the host and how much you can spread. For example, if we take as an example Ebola in human disease, Ebola is very lethal, but it has difficult to spread because it kills its host too fast. At the contrary, if you look at, for example, COVID disease right now, it's not as little, but it can spread much faster and much more far. It can spread more far. So I don't know what will happen to Xylella. Perhaps if it kills too fast its host, it will not have time to be taken up by another insect to impact another tree. So I cannot really say, predict.
Thank you, clear. It's clear your comment. So I think that the first part of this session is over now. I can take some rest and give the floor to my colleague Maria Agnes Jack. So Maria, it's everything is for you.
Thank you, Donato. Hello, everyone. So it's my pleasure for the remaining part of the afternoon to co-chair this session with Donato Bossia. And so now we are going to Apulia, and we'll have a quick trip in Brazil during the remaining part of this afternoon. So first, it's my pleasure to leave the floor to Giusy Datoma. So Giusy has a postdoc position in the Institute for Sustainable Plant Protection in Bari, and she will present her work on the peculiarities of the Dodono strain with regards to its pathogenicity. So Giusy, I leave you the floor.
Hi, everyone. And I'm going to talk about some peculiar biological and phenotypic features of Xylella fastidiosa strain Dodono. Just to remind why it's so important to study this strain. Xylella fastidiosa strain Dodono is the etiological agent of the olive quick decline syndrome. That is a severe syndrome that was diagnosed for the first time in Europe and in Italy in 2013, and so far, according to a recent estimate, affected 4 million olive trees. Also, olive is a new host, so several efforts are needed to gain knowledge about, in general, the disease and about the interaction among the pathogen, the vector, and the host. The molecular mechanism behind the pathogenicity are still unclear. In general, symptoms can be explained by the progressive inability to mobilize water to the upper parts of the plants due to the extensive colonization of the xylem vessels and their blockage by bacterial aggregates, by biofilm, but also plant tylosis and gum. The key point in the virulence is the dual lifestyle of this bacterium, and that means that the balance among an explorative and adhesive phase is the core of the virulence of Xylella fastidiosa. On this basis, the aim of this study was to explore in vitro behavior of the strain Dodono. In particular, some biological and phenotypic traits like cell-to-cell aggregation, biofilm formation, and motility that are also virulence trait of these pathogen were in vitro assessed and compared to those of the type strain Temecula 1 belonging to a subspecies fastidiosa. First of all, the analysis of growth growth curves show that the donno has a slower growth compared to Temecula-1, and also crystal violet assay showed that the strain the donno produced more biofilm and than Temecula-1, and produced a reduced fraction of planktonic cells. So the cell-cell aggregation can be the first step during the biofilm formation. Is the ability to form them to make self-aggregation to form the clumps that can settle at the bottom of the growth support. And if we look at the turbidity of cell suspension in these cuvettes, you can see that the Temecula-1 maintained the same turbidity over 6 minutes, whereas the donno cell suspension greatly changed, and the settling rate that measures the decrease in optical density due to the precipitation of the bacteria aggregates show that the donno has a higher significantly higher sedimentation rate. For this so aggregative phenotype, any attempt to perform experiment in macrofluidic chamber failed, since bacterial aggregates clog the inlet of the chamber, as you can see in this picture. Another feature we explored was the twitching motility. So the twitching motility is a kind of flagella-independent translocation that was demonstrated to be responsible for the upstream migration. And we performed a twitching motility assay on PW and PD3 medium of the two strains, and as you can see in these pictures, the donno colonies lack the peripheral fringe that is associated with this kind of movement, but under the same experimental condition, Temecula-1 produced a large fringe. An important biological trait we analyzed was the suitability for genetic manipulation that can be useful to develop some bacterial mutants that as functional tool to speed up some investigations. In this topic, we have to consider two important points. The first point is that Xylella fastidiosa is naturally competent. That means that it's able to uptake DNA fragments from the surroundings and incorporate them into the genome by homologous recombinations. And the natural competence rates among strains are different, but this ability has been already confirmed and proved for strains belonging to subspecies fastidiosa and multiplex. The second important point is that genome of several strains of Xylella fastidiosa are enriched in restriction modification systems that can impede the acquisition of foreign DNA. And as proof of this, inhibition or deletion of type 1 modification system can increase the transformation efficiency. And we analyzed through a rebased database the genome of the donor strain, of the strain the donor, and also in this gene, in this genome, there are 3 potential type 1 restriction modification systems. For this purpose, we the transformation attempts were carried out using 2 allelic exchange plasmids to develop two mutants, a strain expressing the green fluorescent protein, and a strain knockout for the RPFF gene that is involved in the quorum sensing system. In particular, for the GFP strain, we used a plasmid containing the origin of replication of Xylella fastidiosa developed by Professor Lindo's group. In the other case, we construct a mutagenesis cassette to allow the substitution of the target gene RPFF with the kanamycin resistant gene. In particular, the kanamycin resistant gene among the flanking regions of the RPFF was cloning in a normal cloning vector containing the E. coli origin of the replications. Once obtained these two plasmids, 3 approaches of transformation were investigated. First of all, the natural competence, just putting the plasmid and the donor cells together into a agar plate. But any attempt to naturally transform our strain, the donor strain, failed. Instead, this ability was confirmed for the strain Temecula 1. Then the competence was induced by electroporation, and also in this case, no colonies of the donor were detected on selective medium, and the last approach was to electroporate in presence of the type 1 restriction modification inhibitor. Finally, this strategy was successful with both the plasmid used. The recombination, the integration into the genome were confirmed by PCR. In particular, for the RPFF minus strain, we used 3 primer pairs indicated here by different colors, and as you can see in the photo, in the picture in the middle, the RPFF gene was amplified only from the parental strain, but not in the 3 mutants in the different clone. For the GFP strain, a primer pair encompassing the exchange site was used to confirm the recombination event in the mutant strain. And here on the right, you can see a lot of the donor cells fluorescence that expressing the green fluorescent protein. The findings led us to conduct a sequence pairwise comparison, Temecula 1 versus the donor. of 8 genes that are involved in natural competence and long peeling machinery. And the resulting variations were submitting to the Provean web server that is in order to predict whether these changes can have an effect on the biological function of these proteins. And in silico analysis predicted only 5 variations as deleterious in 3 genes, PIL1, PILM, and COM-AA, but of course, other studies are needed to confirm the variations in the sequence and check for the biological function of these proteins. To conclude, we can say then that the Temecula-1 strain and the donor strain have a different in vitro behavior. In particular, the donor show a more aggregative phenotype and produce more biofilm than strain Temecula-1 that and also has a reduced fraction of planktonic cells. Is not prone to natural transformation like other PAUC strain, and of course mutants were obtained only in the presence of a type 1 restriction modification inhibitor, remarking the critical role of this system in modulating the transformation efficiency. And other studies will be needed to explore and confirm the implanted behavior. So this work was made in the framework of a short-term mission at the Auburn University, granted by Aeroxant Construction. So I would like to say thank you to the construction and also to all the group in Auburn. Thank you very much to Alessandra and all in my group in Italy.
Danilo Vola is a researcher in the Department of Chemistry, Organic Chemistry in Bari. He is a specialist of biohybrid systems using microorganisms, and he will present the work he is pursuing on DSF in Xylella in terms of regulation of pathogenicity factors. So Danilo, I give you the floor.
Yes.
I cannot see you. Yeah, okay, it's great. Thanks.
Thank you. Good afternoon. This is Danilo Bona speaking. I'll be briefly introduce my results in XFACTOR and Fontaine straight collaboration with Dr. Dattoma, Dr. Cicco, Dr. Morelli, Dr. Sardarelli, Saponari, and Professor Farinola, all from Bari in Italy. I come from the Organic Chemistry department in Bari. So I will present my work about the studies on diffusible signal factors molecules in Xylella fastidiosa strain, the Donno. I have some difficulty for... Okay, can you see the second slide? Thank you. Xylella cell cycle is strongly related to the synthesis and diffusion of some signals molecules called the diffusible signal factors, which act in a molecular quorum sensing system. From the state of the art, we perfectly know that bacteria produce diffusible signal factors family. Essentially, they belong to lipid molecules group and family, which are cis2 unsaturated fatty acids. DSF is produced by synthase RPFF and exploits a DSF sensor called RPFC, which are proteins basically, and it helps the virulence enhancement of Xylella fastidiosa, enhancing the ability to migrate and proliferate within xylem vessels in a cell density dependent manner. So basically, we can resume some years of literature about DSFs According to that, Xylella virulence sets on a delicate equilibrium between two phases and forms of the bacterium. One is in suspension, planktonic, motile in xylem, and the one is adhesive in biofilm shapes, which can be resumed as symptoms in the free form that can be carried by vectors, for example, while the clogged system in biofilm that can clog and dilate xylem systems. And all these systems are basically related also to molecular biology, because this DSF upregulates adhesion genes and downregulates motility genes. So basically our purpose is the screening of some chosen commercial DSF extracted from Xylella fastidiosa, Pacua sottospecies, and the new synthesis fatty acid, which is our specialty from organic chemistry department, and investigation on some biological features according to influence on planktonic and biofilm viabilities and molecular biology. Why? Because of a general influence on DSF on biofilm production opens a new therapeutic perspectives, for example. So basically, we started from Xylella cultures and also other control cultures. We optimized solvent extraction of DSF, and we performed also, for the first time, a concentration, purification, and methylation process by using a kit. So we isolated and identified the crudes, and we tested the crudes by a biological point of view. Then we isolated this crude into fractions, and we tested again the same systems. And this one made in Ponte project. So for bacterial culture, we used Escherichia coli, which was able to produce DSF, and Escherichia coli in wild-type mode, and Xylella fastidiosa wild-type mode. And we used the gel light as a physically okay medium, which we could start the extraction, which is able to produce low emulsion and high turbidity with a lipid form, which was enriched by fatty acids. We personally optimized the solvent extraction by decreasing ethyl acetate solvent, the using of ethanol and other eco-compatible solvents for destroying lipid form, and the sanitation as a physical process. And we also moved to specific characterizations like gas chromatography, MS identification, and proton NMR and carbon NMR. And then, thanks to Josie Dattoma collaboration, we moved to some specific biological assays. So basically, as a state of experimental state, we could argue that we used over 130 petri dishes to obtain milligrams of extracts. Then we purified these extracts by increasing the methyl ester fatty acid groups till the obtaining of 45 milligrams after methylation. Then we moved to the column chromatography to obtain the first fraction and the second fraction of high pure fatty acids. So basically, after these solvent-assisted and methylation reactions of the crudes, we could argue that Escherichia coli wild type do not produce any detectable molecules. Conversely, Xylella fastidiosa wild type and Escherichia coli positive to RPFF molecule produce highly comparable GC outputs. And according to the details, although Escherichia coli produced a more complex set of fatty acids profile, Xylella fastidiosa resulted good and more convenient to use as starting material for extracting, isolating, and fully characterizing also the fractions. So the extract crude gave us some interesting biological outputs very in line with the literature. For example, we obtained a strong response of a biofilm behavior of the cells in a dose-dependent manner, and also a strong gene-related, dose-dependent related expression of genes, especially those for adhesion process, so agglutinin like A and B and fimbrial A. So this was in line with literature. And after the column chromatography and the purification of the fractions 1 and 2, in line with the literature and thanks to the NMR characterizations, we fully performed that we fully underline that the found function 1 was a Z2 unsaturated fatty acid, in the specific case analyzed as methyl ester and then an acid, with a single methyl substituent in a alpha position. And again, this is in line with literature. And again, we evaluated in collaboration with Giusy Dattoma the study of this fraction on genes and on directly on whole cells. And again, we observed the trend in a dose-dependent manner of this fraction, both for biofilm-forming cells and also for the expression of genes. So again, agglutinin A, B, and fimbrial A genes. Then we moved to testing commercial amino-synthesis fatty acids. We choose commercial fatty acids which were related to these DSF molecules, a cis-2-desenoic acid, a cis-2-dodecenoic acid, and 11-methyl cis-2-dodecenoic acid, which were respectively signaling molecules in Pseudomonas, Burkholderia, and Xanthomonas campestris. But unfortunately, we did not obtain clear results. And then we moved to the synthesis of new fatty acids, cis-2-tetradecenoic acid, cis-cis-dodeca-2,6-dianoic acid, and cis-cis-dodeca-2,9-dianoic acid, strongly related to some natural substances found, for example, after maturation and fermentation of some fruits. So strongly related to natural fatty acids. We optimize as organic chemists the reaction conditions, exploiting for the synthesis. In particular, we exploited the modification of the Horner-Hammond sulfonation called Stille-Gennari reaction, and we adopted some optimization. We avoided the use of toluene for the dissolution of the dimethylsilyzane, and we used a more green and economical base ligands for similar synthetic approaches, which was the TDA1 instead of Kroneter's. In this case, we observed a different behavior for on biofilm outputs and on the expression of adhesion genes. Biofilm forming cells were decreasing after the treatment of the new synthesis fatty acids. And in the case of the cis-2-tetradecenoic acid, planktonic cells were able to increase in numbers. And there was also an observation of a decrease of gene expression both for agglutinin A, B cells, genes and fimbrial gene A. This was a bit the same trend as the second new synthesis fatty acid, while the third fatty acid was a bit different in the planktonic response due to the decrease of the planktonic forms made in numbers of cells, maybe due to the toxicity, the inner toxicity of these fatty acids. So, concluding, I would like to, in conclusions, we investigated new insights into the biological response of Xylella fastidiosa strain Donno to lipid signal molecules. DSF shares structural analogies with enoic acids and elicits regulatory effects on surface adhesion. And Stridodonno is sensitive to regulation with DSF-like molecules. So we observed significant trends in biofilm, planktonic regulation, and gene expressions with extracted crudes and fractions, and an upside trends for new synthesis fatty acids was observed. The study, of course, needs a broader range of molecules, but it absolutely paves the groundwork for future research into control strategies based on fatty acids modulated regulation of the Donno virulence. Thank you for your attention.
I wanted you to let me know if you have tested any different medium or broth to test if the composition of the broth or the medium has any effect on the aggregation capacity of the Caucas strain.
Yes, we tested. I tested PED3 and PW. Also, I tested to attempt the macrofluidic the experiment in macrofluidic chamber. I tried also with PD2, but the aggregation was very high in all the media.
You think it's an intrinsic capacity of the strain and it's not dependent on its environment?
Yes, I think is a phenotype. So the phenotype is the genotype and the environment. So it depends, of course, of the medium. So implant maybe is another behavior.
Yes, and of course, a link question that was asked was, have you tested bioinformation for this strain, the Dedono strain, using olive sap as a medium?
No, we didn't have extract olive sap, so we cannot use the olive sap. Of course, it could be really interesting to test the olive sap. Yes.
Thank you. Thank you. I've got now a question for Danilo. So Danilo, what do you think about using DSF in biological management?
If I well understand that as a biological management due to like if you refer to biological management as a biological application in the field or the biological application in the lab, I think this is the meaning of the in field. I think this is strongly in line with what we discovered, because of all these materials are perfectly related to natural lipidomes systems. So, for example, it is also this study could pave the way also to the massive production of these fatty acids also from, waste disposal of industries that uses, for example, food grade systems. So I think this could be in principle okay and biocompatible and not detrimental for other types of, species or the same, bugs or flora and fauna both. I think the biocompatibility is very high. And also the type of methodology that could be used for the inoculation could be biocompatible too.
Thank you for this answer. I've got a related question. The last 3 DSF you were using, do you know if they are produced by any member of the microbial communities?
The new synthesis fatty acids?
Yeah.
No.
They are not. There is no natural.
No, they are strongly related to natural products, and this means that the position of double bonds and the presence of one more double bond than the other one is related to. So the family is those of like lipid fermentation, but they are totally new synthesis fatty acids because we have chosen the position of the double bond according to the starting material commercially available. Thank you for the questions.
Thank you. Well, I wondered if you, sorry, it's for Juicy.
Yes.
Juicy, have you tested the capacity of strains to make fringes only for the Dedono strains in comparison to the Temecula one, or did you also tested other Pauca strains?
No, I tested Temecula and Dedono, not other Pauca strains.
So, for example, or maybe you know from the literature or from discussing with Brazilian colleagues, if the CBC strains does make fringes in this type of environment.
So the strain 9A5C is really similar to the Dedono strain, but other Pauca strain has a different behavior. So is not typical of Pauca strains, of all Pauca strains.
So it's 5:05, so I think it's time to come back and to start the second part of the afternoon. So now I think we have already landed in Brazil, and I will briefly introduce Alvesio, who is the next speaker. So Alvesio, everybody knows him. He is well-known in the Xylella citrus area, where he has been working for a long time. He is working in the Citrus Research Center in Brazil. And while having worked a lot on the Pauca strain infecting citrus, he has taken the opportunity during the XFACTORS to study the diversity of Xylella strains infecting olive trees in Brazil. And today, Alvesio will present his work on the pathogenicity of Pauca strains on olive trees. Please, Alvesio, the floor is yours. Welcome.
Okay, thank you for our introduction. I'm trying to share my PPT here. I can find this here. I can't. Oh. That's all right or not?
Not yet, but sometimes it takes a few minutes to. Yes, maybe. It's a little bit transformed. No, it's okay. Now, if you can just put it in the full screen.
Full screen, okay.
Yeah, it will be great.
Okay, that's fine now.
That's it.
Okay.
Yes,
okay. Thank you, Med, and it was a pleasure to attend this conference. Okay, during my talk, I will summarize 3 years of hard working in greenhouse, try to reproduce the olive-acquired symptoms by inoculation. That's kind of symptom that you see in the clear in the field, but try to inoculate different STs from Pauca. This work was done by my undergraduate student, Emily, and by myself, of course, and with collaboration with Alessandra Souza. Okay. Just a moment in that scale. Okay, as Ed shows early, we can classify Xylella by 3 different subspecies. Sorry, that can infect 100 of the plant species, but that can cause a problem in few number of food plant-based food, such as grapevine, plums, olive trees, sweet orange, citrus sinensis, and the coffee. Okay, in Brazil, we have officially reported 2 different subspecies of Xylella, multiplex and the Pauca. But it's Pauca, it's the major threat causing problems mainly in sweet orange, causing the citrus variegated chlorosis, in coffee, causing the leaf scorching, and recently in olive trees. So Brazil, we can say that Brazil is a hotspot of diversity of Pauca strains. You can found different STs in coffee plants, okay, in citrus plants, and also in olive plants. But in olive, even you can find different STs. Only one STs, 16, was represented by 5% of the plants the symptomatic plants in the field was infected by these STs. So the first question is about the ability of these different STs from coffee and from citrus to infect olive plants. Why this the question? Because you can address or you can clear some situations in the field when olive and citrus or olive and coffee are cultivated side by side. Okay, the real question is, there is a cross infection between paucas from coffee and from citrus to olive plants. That is the main questions. So to try to answer these questions, you follow this protocol by protocol inoculation, has screened the bacteria from cryogenic collections, grow the bacteria in PW or BCYE mediums for 6 to 7 days. And also we were suspended in PBS in high concentrations and doing the pre-inoculation in young branches in olive plants. We using different STs and using different two different olive varieties, Grappolo and Manzanilla, and also different plant sizes. All the tests that the efficiency of inoculation was did by traditional PCR using RST primers. So here you are summarizing 5 different experiments during conducted during almost 3rd month. You can see that from the 10 different Pauca STs that you try to inoculate, that you inoculate the plants, at least 9 different STs from olive plants, from coffee, and from citrus sinensis. All of them could infect olive plants. But it's interesting that even you one ST classified as Pauca from Prunus, inoculate from Prunus, and also the ST from the subspecies multiplex that was, of course, isolated from Prunus, both could not infect olive plants in greenhouse conditions. So you have here different frequencies of the infections in olive plants in the, from Pauca strains, independently if the strains comes from citrus, comes from coffee, or of course comes from olive plants. But strains from plums could not infect olive plants. But unfortunately, there were no systemic establishment of the bacteria in the host, and consequently, you can't observe any symptoms associated of all quick decline in olive plants. So, based on that, we changed the procedure of inoculation. But based on the hypothesis that the choice of medium, rich medium or minimal medium, could alter the bacterial genes important to plant colonization and pathogenicity. So it is an ongoing experiment. The method was you grow the different STs in PD3 mediums. After that, we collect the cells, the colonies. Okay, we standardization the bacterial concentrations in PBS. Okay, and after you put it in on fractions in XFM medium, in another fraction in PD3 medium. We stay that in this liquid medium during 24 hours, and again, centrifugation the bacteria and the padronization, the concentration of the bacteria, and pricking inoculation in different hosts. Here, you're using olive, citrus, and tobacco plants, and also the bacteria was checked for inoculation using the RST primers. Based on the results from the olive plants using Xylella, using different STs of Xylella, this is a summary of the results that you can see here. you're using different STs, you can see that there is variable results from if the bacteria was kept in PD3 or XFM medium. For instance, for some ST, you can see some better colonization of the plants when the bacteria was kept in XFM medium. And for another strains or another ST, you can see, for example, over here, you can see the bacteria from hibiscus that came from that was kept in PD3 medium was able to better inoculate the plants than colonize the plants than the bacteria that was grown in than the bacteria was grown in XF medium. That result was obtained 180 days after inoculation. So in overall, you can see here that the bacteria that was kept in XF medium was able to colonize more with more frequency the plants, the olive plants, when compared with the PD3 bacterium. When compared with the bacterium that was kept in PD3 medium, but you have no statistical difference. But unfortunately, we have no symptoms in positive plants, in olive positive plants yet, after 204 days from inoculation. Okay, but when you're looking for the data from the citrus plants, you can see a little bit different. You have a high efficiency of inoculation in overall results. You can use 4 different STs with a lot of plants that you have been inoculated. In general, you can see that the bacteria that was kept in XFM was efficiency was almost 89% compared with the 65% of the bacteria that was kept in PD3 medium. And you have one statistic closer to 9.5% of the significance. So it's interesting that the symptoms, the history was quite different. The bacteria that was kept in XF medium was more results in more symptomatic plants when compared with the PD3 medium. Okay, you can see here a high different level of the significance, and also you can split the different severity of the bacterium when you are comparing XFM or PD3 medium. And also, just to remember that these results come here, it comes from 204 days after inoculation. Now, when you are using tobacco plants, has more tobacco plant. We know that's a model plant to work with Xylella fastidiosa. We can see no difference between if the bacteria was kept in PD3 or XFM medium. It also have no difference between the 2 different STs that you use in ST13 and XT84. You can analyze this data 9 days after the inoculation. Regarding the symptoms in tobacco, also independently of the medium that the bacteria was kept, we have symptoms in olive, like the classical symptoms that you can see, that's brown spot on the leaves, on leaf surface, independently of the strain or independently of the medium that the strain was kept on. And also, you can see a huge difference between an inoculated and no inoculated plants, okay, by the plant size. So you can see, so the no-inoculated plant, the plant was tall, much more tall than the inoculated plants. But again, you have no difference between the ST and the medium that you're using regarding to tobago symptoms. As you know, tobago is a plant, it's a model plant to work with Xylella, and we will not expect different results because it's an easy plant and high susceptible plant to infections. Take together all these results that you have obtained until now, you can say that the passage of the bacteria in minimal medium, XFM improved the infection efficiency of resilient plants for infection, such as olive and citrus plants. The CBC symptoms was significantly increased by the passage of the bacteria in XF medium, that you are investigating what the genes that could do start on or down regulated with the passage of this bacterium. And for now conditions, the latency of olive quark symptoms in greenhouse conditions seems to be longer, mainly when Xylella fastidiosa is artificially inoculated. Why? Because parallel, you do some experiments, try to investigate the vertical transmission of Xylella by routing infected but asymptomatic spores. Okay. You take went to the model in the field, you certify that this model was infected by Xylella and it has no symptoms. So you propagate these spores. And you wait for almost 2 months, 3.5 years in the greenhouse to see the olive white symptoms. Okay. The classical olive white symptoms. So the latency for us, the latency of olive white symptoms, it's longer. It's longer than citrus, and also in the field when you talk with the growers, it is reported that the symptoms can see in the fields only with plants with 3 or more years old. Okay, that's all. Thanks for the attention. I'd like grateful for the XFACTORS for the grants necessary that was conducted this experiment, and also to São Paulo Research Foundation, FAPESP, that provided the grants. Okay, thank you.
Thank you very much, Alessio, for a very interesting talk. Annalisa, she's a researcher at the Institute for Sustainable Plant Protection at Bari. She's working with Maria and Donato. And she has been involved in the study of Xylella since its discovery in Apulia. And today, she will present her work on olive response to multiplex and Howa strains. So, Annalisa, the floor is yours.
Thank you, Maria-Agnes, for the introduction. So the topic of my talk will be the transcriptome study on the different plant response elicited in a susceptible cultivar inoculated by different strain, so 2 different strains, a Howa strain and a multiplex strain. So this study was prompted by this situation. So we know that olive trees in Europe were found infected by different genotypes of Xylella, in particular after the detection of ST53 in our region in 2013. Other ST, as ST80 and ST81, were detected in olive tree, cultivated and wild in the Balearic Island. In 2018, the sequence type ST6 was also detected in olive in Madrid and then was eradicated. Even though ST6 was mainly associated to almond-causing almond leaf scorch disease in Alicante, whereas ST6 has not been detected in olive tree, we were asking if olive could be potentially become an host of ST6 genotype. And to answer these questions, in Spain were started some pathogenicity experiment on different olive cultivar with different ST genotype, including ST6, and until now no symptoms were observed. Moreover, they found a low concentration of bacteria in the inoculated plants. But for more detail on this work, I invite you to read the poster presented for this conference by Miguel Roman from CSIC. Again, no leaf symptoms were observed on olive plants inoculated by ST6 strain in this experimental test published in a paper by Nane in 2019. So starting from that, we were asking which is the olive response of ST6 in a very susceptible cultivar as Cellina. And so to answer this question, we started 3 years ago artificial inoculation in a greenhouse condition using ES-well strain on Cellina olive plants. And as you can see, after 11 months from the inoculated, we observed only in plants inoculated with the Donno strain, the ST53 strain, which we used as a positive control in our trial, we observed just a down rolling on the leaves, followed after 19 months by complete desiccation of branch, as you can see here in more detail in these two pictures. So no symptoms were observed at the time on the plant inoculated by ES-well, and also RNA was extracted from the xylem of these plants to start transcriptome studies. So after 3 years, the situation on olive inoculated by the Donno you can see the plants are completely desiccated and some plants are dead, where olives infected by ES-well are starting to show only a specific symptoms like delay in sprouting and leaf chlorosis on some shoots. This situation is in agreement with the lower titer of bacteria corresponding to this value of CQ found in a plant inoculated by ES-well compared to the plant inoculated by the Donno. And now I'm going to show you the results obtained using the transcriptome approach. So 9 libraries were constructed from 3 biological replicates for 3 conditions that from now I call MOC, DD, and ES-well. Raw reads after the quality check were mapped on all the 3 olive genomes that were released over the time. And I can say that the best rate of mapping was obtained with P-qual genome, and the relative count dataset was used for the subsequent analysis. The sample-to-sample distance analysis show a good correlation among the biological replicates and also a clear separation of DD samples from the MOC and ES-well samples. Principal component analysis on samples also clearly grouped the ES-well sample close to the MOC according to the principal component 1, which explained the 48% of the entire variability. This represents a nice result, also confirmed when we did a principal component analysis on all the sample, on all hernia-sac sample that we have available from our all our inoculation trial, including other cultivar and other strain, as the strain CO33, a sandy strain, which we know is able to affect the olive plant in artificial inoculation trial. Before the differential gene expression analysis, we also explored the principal component analysis by hierarchical clustering analysis, the top 100 most variable transcripts, and we obtained 3 groups of transcripts. In the first group, there are transcripts with higher expression value only in the ES-well sample compared to the MOC and the Donno, and most of them were functionally annotated as expansine like B1. In the second group, there are transcripts with a lower expression value only in the Donno sample compared to the MOC and the ES-well, and these transcripts are well known to be expressed in the vascular tissue, so involved in cell wall mechanism. In the third group, instead, you can see there are transcripts that are upregulated in the Donno samples, and these transcripts are transcripts well known to be upregulated in the PTI response against the pathogen. So this last group was not upregulated in the ES well sample. So now let's see to the results from the differential gene expression analysis that we performed on the all 3 genome count dataset, and consistently among the 3 genome we obtained a much higher number of DAGs in response to infection by the donor compared to the number of DAGs from ES well versus MOC. So we moved just to compare by Venn tool the list of DAG up and down regulated with which have the higher fold change value just for the P-equal genome dataset, since in Farga most of the DAGs were lacking functional annotation description. So then next I'm going to show you just the genome ontology enrichment results obtained from the upregulated DAGs categories. And as you can see in the category index that are upregulated in both conditions, there are geoterms enriched. The geoterms, these genes are enriched for geoterms related to defense against bacteria. So it means that both condition, the plant sense the presence of bacteria. But if you can see at the DAGs in plant infected only by ES well and upregulated specifically to this condition, these DAGs are few and are also related to expansine again, as we saw before. Whereas this is the specific response in plants infected by the donor. So with several geoterms enriched for defense response, cell wall modification as callose deposition. So you can see how much the 2 responses are different against, so the plant response is so different against the 2 strains in the same cultivar. After obtaining these results, we were asking if this difference was due to some difference in genomic composition between the 2 strains. And just for the talk and hopefully to receive some suggestions from you maybe, I have added 2 slides with some our observation and speculation, I would say, on them. So first, comparing the gene content of the 2 genomes, we found an O antigen acetylase gene, which is present in the donor but not in the ES well. This gene is already known to be present in the Pauca strain to be involved in the acylation of carbohydrate moieties on the extracellular acetylase molecules of LPS. And moreover, we know also that the O antigen is well known to be important for Xylella to delay the plant innate immune recognition. This gene, the gene product, I have seen that contain these 2 domains, and both domains are well described in another pathogen, so in the Salmonella enterica, in a recently paper reporting the mechanism of acylation of antigen, and reporting also that this mechanism affects important features for the bacteria as immunogenicity and virulence. So we were asking if this lacking of this gene, it could be correlated to different response of plants against the 2 strains. And another gene that we found is this, so it's a gene encoding for an enzyme belonging to alpha-beta hydrolase superfamily, again only in the donor and absent in the S well. This finding seems more interesting since it was present not only in Pauca strain but also in CO33 strain, a Sandy strain, which, as I told before, is known to be is able to affect the olive in artificial inoculation. Moreover, the gene product of this gene was already reported in this paper as present in the vesicle prepared from 2 Pauca strains. And so again, we were asking which is the involvement of this protein in different pathogenicity observed of the 2 strains, so in the donor as well. So still many questions are rising, but now let's move to the conclusion. Okay, so olive is one of the host species infected in Europe by different genotype, including ST6. ST6 is mainly associated to almond leaf scorch disease, and even though it's not detected in all of tree, except for one outbreak already eradicated, we start to test if olive could become an host of this ST6. We inoculated olive plants of Cellina cultivar, which is very susceptible with S1 strain, an ST6 strain, and no symptoms were observed after 3 years from the inoculation, compared to the severe symptoms from control. And we performed also a transcriptomic analysis on this plant, as we found only in plant infected by the donno. The transcriptome was drastically reprogrammed. Therefore, we can say that the comparative genomic and transcriptomic approach are providing useful insight for exploring the molecular basis of Xylella olive interaction. To finish, I want to thanks my colleague from my institute for their hands in this team's work, and I want to thank Blanca Landa and Esther Marco-Noales from Spain for the nice collaboration, born during this project, and obviously the XFACTORS project that I personally define as a journey from resilience to resistance. And thank you for your attention.
Let's go for Elvesio first. So Elvesio, there was a question for you regarding ST53 in Apulia. So Maria said that they found that vector-mediated inoculations were much more effective than needle inoculation. So do you think that it can be needle inoculation per se that is not an efficient method to inoculate olives? Do you have any experience with vector transmission in olive in Brazil?
No, I personally don't have, but João Lopes, maybe he'll talking tomorrow afternoon, and you are talking about vector transmission of Xylella in olive plants.
Okay. Thank you.
Because just explain you the project you're split in two different ways. I'm working with the bacterium, and João Lopes was working with the vector.
For sure, it is often the case. Yeah, sure. Thank you very much for your answer. So, Annalisa, there was a question for you. Giuseppe Stancanelli from EFSA said, I understand that you could infect and cause symptoms in Cellina di Nardo with multiplex ST6. If this is confirmed, can the different olive response to different subspecies and STs observed in different areas be more linked to olive varietal susceptibility than to strain virulence?
So if I understand the question, we performed the pathogenicity experiment with a very susceptible cultivar. So the Cellina is the very susceptible cultivar to Xylella, and you have seen the bacteria is sensed by the plant because we found some CQ mainly in the initial part of the experiment. The CQ of also for the ST6 we observed the presence of bacteria, but after the time it seems that ST6, the multiplex in our cultivar in Cellina decreased its concentration. And I don't know for the other cultivar, maybe Blanca can say more than me about the test and using other cultivar. But we choose this cultivar just to facilitate the pathogenicity of Xylella, because we know that Cellina is a very susceptible. I don't know if I captured good in better way the question.
I think so, yes, sure. I've got a question also for you. You observed that the O antigen acetylase gene was, I don't know if it was absent or if it was mutated in the multiplex strain in comparison to the Pauca strain. But do you know if this difference?
No, the difference is the presence. So this gene, you can hear me? So
Yes.
these genes, the protein of this gene is completely absent in the as well, in the multiplex, and is present in the donor. This comparison comes from the alignment of the scaffold, of the genome scaffold. So I'm sure that the protein is, the gene coding for this protein is not there, and also is not in another other position of the genome, because we did a blast on all the genome, the blast P of this protein. So this gene is not present, is not mutated, but is not present at all.
And do you know if this gene is also absent for all other multiplex strain, or is it present?
Yes, I'm expecting this question. So I have also another slides where I collect all the genomes. It seems this is a PAUCA, is a typical PAUCA gene. So it's present in PAUCA, but not in multiplex.
Okay.
If I can share again, I have a table, but I don't know is it possible.
Okay, there are another question for you. Marta Robledo is asking if you try to map your RNA sequence against the corresponding Xylella strains that was inoculated.
The map, the reads, so the reads comes from plants, mRNA, so we don't get reads from bacteria transcriptome. I don't know if
you couldn't isolate bacterial RNA from these plants.
We enrich for the plant mRNA, not for the bacteria.
Okay, so you didn't test on the bacterial RNA, so you cannot answer a question. Okay.
Yeah. Maybe it's a good idea to test also the presence of genes looking at the RNA of bacteria in plants. This is a good suggestion.
Yes. Elvis, I've got a question for you. You apparently have a very large diversity of strains that are infecting olive in Brazil. And all these strains, are they causing an economically important disease on olive over there, or is there just a few trees that are affected, but altogether it's not an epidemic situation?
No, it's an epidemic situation.
Okay.
Okay. It's the symptoms are severe symptoms you can observe in the plants, and by the time the plants die also. But in general, you can see just one ST that is prevalent in a few ones that it was more like it's not so. It's okay. Occasional occurrence. Okay, but the mainly ST it's ST16.
Yes.
That was first reported in coffee plants. Okay, but it's a epidemic situation. Yes.
Okay. Thank you very much. I think, Elvis, there is a question from Emilio Montesinos. He asked, you inoculated 10 to the 7th CFU per mil with a DO of apparently 1.2 to 1.4. So in your experience, the OD values correspond to much higher cell concentration. Is there any difference among strains?
Could be. Thanks, Montesino, for the questions. I will check the number with my student, but I could be, but I could be by the strain, of course, because the PAUC strains grows more, I can say aggregate, and you do the CFU. It's not so a good correlations between the OD and CFU.
Yes, of course.
But thanks, I will check the numbers.
So it's my pleasure to leave the floor and to present a recent work on the phenotyping of olive response in order to estimate Zalella load, which is a very interesting and appealing proposal. So I leave you the floor.
Thank you, Maria Agnes, and good afternoon again. I hope this time you can hear me better than before. Yes, the part of the work I'm presenting is just a part of the whole investigation we are doing as a team in Bari for the resistance, for the characterization of the resistance in olives. I will present more the part of the field and the diagnostics, and the following presentation by Pasquale will be more on the molecular part. So our question for this experiment were if considering that we had field evidence of different susceptibility of the olive cultivars, if this impacted also the prevalence and the rate of spread of the infection in the orchards made where different cultivars are grown. And also we wanted to see if we could confirm that in the cultivars with resistance phenomena we observed, we could see a differential bacterial population size. So if there was any correlation between the symptom severity and the bacterial population, as well as to understand if in trees of cultivars showing different susceptibility, if we had a different distribution of the bacterium within the plants and within the tissues of the plants. To do this, we worked in the infected area. So we worked with the plots, commercial plots under natural pressure of inoculum. For this reason, we were limited in the cultivars that we could analyze and we could focus on, because we had to rely on mature olive trees grown in the area. So we could select in one area plots of these 5 cultivars. From the left to the right, we have the Oliarola and Coratina, which appeared more susceptible to the infection of Xylella fastidiosa ST53. And on the right, those that so far are known to be resistant, the FS17 and the Lecino. And in the middle, one cultivar, the Frantoio, which based on our observation appear to have an intermediate behavior between the highly susceptible and the resistant cultivars. And we tried to choose these olive orchard all in the same area, and all of approximately the same age. So we could say that these 3 were there since the start of the epidemic. So they were from 20 to 30 years old. Of course, we could not have exactly plant trees of the same ages. And these are the results regarding just a survey in this plot for these 5 varieties. The number of plants is different for each one, because, as I said, we were limited to what we could find in the commercial plots. But as you can see, clearly the 3 first listed varieties had a very high incidence in the plot of in each orchard of infection, compared to the resistant cultivars, in which the infection rate in the orchard reached approximately 50–60%. Some of the infected trees were monitored also for symptoms for 1 year, and in the last column you see the progression of the symptoms. I have to say that for Oliarola, when we started the experiment in 2019, we were already at the maximum expression of the symptoms, with 4.5 a score on a 5 value based. And whereas for Coratina and Frantoio, as you can see here, we observed an increase of the severity of the symptoms, as shown in this picture. For example, is the Coratina, for which during the year of observation we observed this suddenly decline that occurred on the infected plant, as well as some severe infection, some severe desiccation also on Frantoio, whereas the situation appeared quite stable for the two resistant cultivars. Besides these commercial plots already existing in the area, within another experiment, we set up two commercial plots, one in 2018, one in 2019, by planting plants of the same age and by alternate rows. So we had one row of the susceptible cultivars, which in this case were Arbequina and Arbosana, and one row of FS17. And you can see that when in last month we did a diagnostic survey, how different are the incidence of the infection in the same plot. So we reached, after 2 years from planting, the one planted in 2019, 26% and 2% only for the resistant cultivar. Whereas after 3 years of exposure of this plant to the infective vectors in the area, we reached a percentage over 50%. You can also see how without any good management of the vector population, you can see how the incidence of the infected plant can doubled from 1 year to the other. And in the plantation made in 2018, this year we could already start to see some initial symptoms of desiccation that are typical of the Xylella infection in olive. Of course, not all the infected plants were symptomatic, but the initial symptoms could be seen on some plants. Then to see the correlation between the cultivar response and the bacterial population size in the plants, we selected for the 5 cultivars that I mentioned before, 12 plants which were followed which were sampled 4 times during 2019, as you see here, and we did the test separately from on the small branches, on twigs, and on leaves. And these are the results of the detection in these plants. As you can see, we had an efficiency of the detection close to 100% in the 2 highly susceptible cultivars, Oliarola and Coratina, regardless of the period. To our surprise, the end of summer in olive, we got the lowest detection efficiency, while we did not expect these results. Whereas going through to the resistant cultivars, you can see that the efficiency of detection decreased, and the tissue where we could have the highest detection were twigs, and that despite the period of sampling allowed to gather the highest value of detection. And I have to see that it's particularly important to remark that in the resistant cultivars, especially in the FS17, we detected a very low level of bacterium in the leaves. When we checked the bacterial population size in these plants, we estimated by an indirect measure through qPCR assay, we got similar results as the detection efficiency. I mean, as you can see in this graph, we could statistically differentiate the 2 resistant cultivars for having hosting a lower bacterium than the susceptible cultivars. This was consistent in all sampling period, with the cultivar Frantoio, which is the one in green, showing also here in this evaluation an intermediate behavior. In some period it grouped with the susceptible cultivars, in others with the resistant cultivars. It is interesting to see that we, for the diagnostic efficiency, here we have consistent results also for the population size in the plants regarding the period. With September, as you can see here, in September, we detected the lowest population size in all the cultivars. So the results were consistent with of course, with the efficiency of our detection. And the results showed before with an indirect estimate of the bacterium. We did also isolation in pure culture, and just as an example, you can see here the plate obtained from the stem imprints made from Oliarola. We're almost in 100% of them you can see the white colonies, while for the S17 only in few spots we could see active growing cells of Xylella fastidiosa, confirming the lower population size in this tissue. Then we focus regarding the tissue. We focus more on the leaves, because we know that the leaves have then, and the young tissue, the young apical shoots have a role and have an impact on the vector transmission. You will hear about this more tomorrow by my colleague Vincenzo Cavalieri. But again, here in the leaves, we have in the resistant cultivars a significantly lower bacterial population than in the susceptible cultivars, and this you will see will have an influence in the transmission efficiency. It is not the bacterial population size, but we found also that taking multiple samples from a canopy, testing multiple branches, we have a completely different situation between high susceptible plants, trees of the cultivar, for example, Oliarola or Coratina, and those obtained from Lecino, the FS17, showing that the bacterium in most of the branches was either absent or I can say not detectable under the limit of the detection threshold of the qPCR that we used to monitor and to assess the presence of the bacterium. And these results are also the fact that we found that the twigs in olive were found to be the most suitable tissue for detecting the bacterium throughout the entire year is also confirmed by the evaluation of the population size of the bacterium in the different tissue. With, as you can see in this analysis, twigs showed the highest value of population size in all cultivars, and again, as showed for the leaves, the population size in most of the tissues was significantly lower in the resistant cultivars. Then we had the opportunity to monitor the population size in olive plots of the resistant cultivar. In particular, in this case, was for the selection FS17 under different management conditions. We had always worked under good management condition, but we had also the opportunity to monitor some poor managed groves, in which, for example, as you can see in this picture, we could observe this important phenomenon of desiccation that, even if limited to few trees, were, I can say, alarming when we first discovered this. And so what we did is that we sampled several trees which did not show symptoms or had a mild desiccation, compared to these trees that of the resistant selection that were showing symptoms. And the results were quite clear and with, as you can see on the right part, almost all samples taken from the symptomatic FS17 trees showed thank you, Maria. Showed high concentration of the bacterium. The level of 10 to 5 CFU is a sort of borderline. What we find in generally in the susceptible cultivars is between 10 to 6, 10 to 7, and 10 to 5. Whereas if you can see in the results obtained from the trees that did not show symptoms, only in less than 6% of these trees we could find this concentration of the bacterium, while the majority, more than 80%, had harbored a low concentration around 10 to 3 CFU for ml. So we found this positive correlation between symptoms and bacterial population, even in the resistant cultivars. So to conclude, this analysis based on field and diagnostic activities show that in olive, the bacterial population correlates with the phenotypic response. The bacterial population and the prevalence of the infection in the resistant cultivars was consistently lower than those observed in Oliarola and Coratina, regardless the period and the tissue. We observed also that in situation of poor management, the population size can increase also in resistant cultivars, being correlated with the symptoms severity. We confirmed that not all cultivars susceptible to Xylella the susceptibility in olives can range from high susceptible to medium susceptible, and maybe Frantoio is one of these cultivars which has this intermediate behavior. From this experiment, we think that we got also important information for the detection of the bacterium in olive. We found that using the twigs can improve the efficiency of the detection. And also surprisingly, we found that sampling not only in the standard period, which is summer and early autumn, can get good results, but also for olive, also winter was shown to be a good period for sampling. And in particular for FS17, it was very remarkable, the lower rate of infection of the bacterial titer we found in the leaves, and this should be considered when doing the monitoring in area where different varieties with unknown resistant behavior are present. Thank you very much for your attention.
Pasquale is well known for his work on olive resistance or tolerance to Xylella. And today he will focus his talk on the role of plant cell wall in the resistance of olive to Xylella. Pasquale, the floor is yours.
Thank you, Marija. I hope you are not so tired. I will give our insights on the key role of the plant cell wall in the olive interactions with the Xylella. Sorry. Okay. Okay. Our background information. Sorry, I have this one. Our background information starts from the Pierce disease, and we know that this from all the studies that were done in this system, that Xylella has a battery of hydrolytic cell wall degrading enzymes. The needs of these enzymes for the bacterium is to allow to move from one vessel to the adjacent vessel, and in this way to get systemically systemic colonization of the plants. But to go across one vessel to another, Xylella should move through some porous structure, which are pit membranes, which normally, when they are intact, they do not allow Xylella to pass through. But Xylella needs to degrade this pit membrane by these hydrolytic cell wall enzymes, and in particularly the pectic and also the cellulose components of these pit membranes. And this is demonstrated by the existence of cell wall degrading enzyme mutants of the bacterium, which are impaired in xylem colonization and symptoms, as you can see here, for example, for polygalacturonase mutants or chitinase mutants. And then this pit membranes degradation was already known from this work from Sun et al. In 2011, and you can see here the progressive stage of degradation of the pit membrane in this interesting scanning electron microscopy photo from Sun. These authors also observed that the percentage of degraded pit membranes was higher in susceptible grape varieties with respect to the resistant cultivars, which have a low rate of degraded pit membrane. This study was continued and published recently by Engel in 2019. And this author showed that a more susceptible cultivar, Chardonnay, is a nice substrate for the Xylella, because it's a good substrate for the cell wall degrading enzyme because of the pectin and composition, and also the cellulose composition. And also in this system, these authors demonstrated that mutants in 2-endoglucanase, which particularly degrades cellulose, they were impaired in colonization. So they introduced a concept that the plant response depends on the cell wall composition on this. So our question was, do we have evidence of this pathogenesis mechanism in Olling is involving in the cell wall? Our model system are chronic infections, that means 1 year post-inoculation infections in greenhouse conditions of resistant cultivars Lecino and FS17 and Cellina dinardos susceptibles. Plants were inoculated with 3 different strains, CO33, subspecies Sandy, our Dedonno strains, subspecies Poca, and the ESVL strain multiplex that you have learned from Annalisa Gianpetruzzi before me. And we approached this study, addressed this question by transcriptome profiling and by microscope observation, considering in transcriptome profiling different combination of cultivars and Xylella strains. As a general overview of transcriptome analysis, we are perfectly able to distinguish the infected plants according to the infectious status, to the presence of Xylella, or also to the cultivars. And here you have an overview. Sorry, here you have an overview of our system in potted plants, in which we observed symptoms in the combination of Cellina with CO33 and Cellina with the Dedonno strain, whereas we did not observe symptoms or very limited symptoms in Cellina, in Lecino with CO33, which was a confirmation of our observation in the donor, in FS17, and also in Cellina with ESVL. Another piece of information that we looked at, and it is needed for our studies, is that either the donor strain or the CO33 strain have potentially functional polygalacturonase gene. From our previous transcriptome profiling, we observed that a sort of cell wall integrity sensing system is perturbed during Xylella infection. And this is because we observed upregulation, and particularly in Lecino, of some leucine-rich repeat receptor-like kinase, which are on the surface of plant cells. An in-depth studies on this system, this infection with strain CO33, and particularly an evaluation of the de novo assembled transcripts from this RNA-seq dataset, told us that an additional categories family of protein, which are called wall-associated kinase, were upregulated in Lecino with respect to Cellina. And this category, these proteins are, of course, are receptors, and are receptors particularly associated to the binding of pectin and oligogalacturonides. Oligogalacturonides are components, are degradation products obtained after the polygalacturonase degradation of the pectin of the cell wall. So a so-called damage-associated molecular patterns role in Xylella interaction in olig. After that, we look at all our transcriptome RNA RNA-seq dataset, all our information, our dataset that we have in the lab, all from greenhouse infections. And we observed that a couple of wall-associated receptor kinase, this category of proteins, was overexpressed in Lecino, infected by CO33, and in FS17, infected by the donor. The protein is not completely absent from, not completely upregulated from the other strain, but is, I would say, moderately upregulated with respect to Lecino and FS17. And when we look at the gene structure of these two genes that were upregulated wall-associated kinase, in this case from an RNA dataset from PQAL, we observed that these genes were truncated, do not express the complete domains necessary to express complete and functional wall-associated kinase. While when we observed the transcript de novo assembled from Lecino, we observed that these transcripts, they encode for a complete protein, possessing all the domain necessary for a functional proteins. This situation was also present in Cellina. The transcripts that we reassembled from Cellina and that were annotated as wall-associated kinase, they were interrupted from because of the presence of an intron. Such an intron is also present in the transcripts from PQAL that we got from net, from the database present on the net. So we are investigating this intron retention mechanism regarding the expression of this gene. Of course, we looked at the expression, at the alternative expression of this gene, WOC gene from Howa europea, we call OE-WOC from Howa europea, in other set of plants, other greenhouse-grown olives chronically infected by our Apulian strain XFDD. And we were able to confirm the overexpression in Cellina and in Lecino, even if we were not perfectly able to associate this upregulation, this increased expression of this gene with the Lecino resistance. That's the other part of the story. We looked at by microscope observation to find such evidence of cell wall involvement. And here you have a microscope, normal microscope observation of olive, in which we distinguish vessel, xylem vessel, from fiber tracheids, which has more a structural meaning. And I do this distinction because while in vessel-to-vessel pit membranes that interconnect vessel to vessels are bordered pit, so you see here the pit membrane that connect vessel to fiber tracheid, they are half-bordered pit. And I do this distinction because we found Xylella in Cellina, in olives, only, as you can see, in vessels. We did not find Xylella in fiber tracheids. And when we found Xylella, in this case, you can see which is moving through a pit membrane interconnecting two vessels, we observed that the pit membrane which constitute the porous structure was degraded as an uneven texture of the pit membrane. While if you consider the pit membrane here is really clear from an healthy plants, you can see by yourself that is very intact. So our conclusion are this one. Do we have evidence of a cell wall involvement in olive? We think yes, we think we have this evidence because we have evidence from molecular science, so and also from the transmission electron microscopy studies that indicate that the cell wall involvement is at the base of Xylella olive interaction. Can it be, is this a pathogenesis mechanism in olive? We think yes, it is a pathogenesis mechanism for the same reason, and also because both strains that induce this is corroborated by the fact that both strains possess a battery of cell wall degraded enzymes that are able to degrade polysaccharides component of the cell wall. And the last question is, can this be part of a mechanism associated to olive resistance to Xylella fastidiosa? It seems yes. We need further studies to decipher the gene expression regulation of this Howa europea wall-associated kinase, and also we should look at the cell wall composition in resistant, chemical composition in resistant and susceptible cultivars, which are ongoing. So this is, thanks, I'm talking on behalf of all my colleagues, Annalisa, Raed, Giussi, Angelo, and Maria Saponari, and thank you to all for attending this presentation.
asking this question, but the person was wondering how you calculated the bacterial load within the plant material.
Yes, this is very tricky for olive because we cannot isolate from olives, so it's very from the leaves of olives, so we cannot do like a serial dilution. So what we did is that for our strain, we made for our strain we evaluated in vitro the CFU based on the OD at the spectrophotometer, and we established that 0.5 OD are approximately correspond to 4 times 10 to 8 CFU per ml. So when we did the test in the plants, we compared the spiked samples with this concentration with the standard amount of cells, and so we retrieved from a standard curve this the quantification. So, but yes, it's very tricky, and is I would say an estimation. But it was clear that there were difference, even if it's not an absolute exact number of CFU. It was clear that there were difference between the resistant and the susceptible cultivars.
Thank you for this precise answer. There was a comment followed by a question. So, can you really call cultivar Lecino and FS17 resistant ones? Bacteria are still propagating, so can it be better to call them tolerant? And can those trees contribute to spread the bacteria by vectors?
Yes, we call the, I have echo. We call the resistant because they counteract, they restrain, they limit the bacterium in the plants. Whereas for tolerant, we mainly indicate a plant in which the pathogen can sustain a normal multiplication and colonization, but phenotypically doesn't show symptoms. So in our case, the plants counteract the multiplication and the colonization of the bacterium, so we call the resistant. Yes, these plants can contribute to the spread of the bacterium, but at much lower rate. As you will see, I said tomorrow Vincenzo Cavalieri will show the results of the vector transmission test using the resistant cultivar.
And I have just a short question before we go to speak to Pasquale. You were mentioning some management practices that favor bacterial multiplication and symptoms in FS17. Can you just briefly mention what are these management practices that are really disfavorable to olive?
Yes, for sure. The irrigation was very important, pruning, and so keeping the plants clean from the desiccated branches. But irrigation was very important. I think was the important aspect that differentiated the two orchards that we were observing.
Okay, thank you very much. So Pasquale, there are several questions for you. So one was, would you expect that if you infect olive with PGLA mutant or KA mutant, or a WAC is not upregulated?
I think so. Yes. Also because last week I heard, but we will. I heard of a seminar from the group of Silke Robacek, and they also looked at this WOC2 in Arabidopsis, which is particularly responding and binding to the oligogaratturonides. So I think it should be this way.
Okay, so you find a strong relationship between expression of this WAC and resistance. Can it be a way to select For resistance in a
Blanca,
faster way.
Blanca, it would be a dream. We are looking for some markers to screen varieties. I don't know. That will be also in our conclusion. It will be also we should do transgenic knockout olive to verify this.
Yeah, sure. And there is an intriguing question. Do you think that Xylella infects only the vessels because it can go to the fibers due to the morphology of the pits?
Yes, perhaps it's the morphology of the pit. They are half-bordered pits, and the pore, the pit is not completely accessible. It finds perhaps the cell wall degrading enzyme are not able to degrade the last barrier to go into the fiber tracheids because of the different chemical composition.
Yeah. Okay, and I just wanted to know if you have noticed any relationship between the size of the vessels and the tolerance susceptibility, as it was shown in grapevine also.
Yes, that's we are starting to think that resistance is a multifaceted phenomenon, which is true also for grapevine, as far as I learned from all the Pierce disease study. There is a poster from Montilon and other colleagues that relate the size of the vessel with the susceptibility or resistance to the bacteria.
Yes. So I think you will speak a little bit about resistance later on in the concluding session. But there is another question for Maria by Wopke. Wopke Wanderwerf asked if he did understood well that you said that you found a decline of Xylella prevalence from one year to the next. And if it understood well, then how is this possible?
No, maybe the slide, the table was not very clear. They were two different plantations, so one was started later from the other, so they were not related.
So maybe Donato, I leave you the floor if you want to share some interesting remarks regarding this afternoon talks.
Okay, I have some remarks to share. I don't know if they are interesting or not.
Of course,
I was thinking a little bit about what has been reported by our French colleagues at the beginning of in the first two presentation of the session. So regarding the estimation of the year of introduction in Europe in general, so in Italy, in France, and but if we looking also to the results of the project published before also in Spain, so similar situation. So in my opinion, the observation of these results obtained during the project is the learning that the introduction of Xylella in Europe is, yes, relatively recent, but less than previously thought, at least according to my knowledge. Less than previously thought, I don't know, 7-8 years ago when after the first detection in Apulia. This means that the process of adaptation, establishment, and spread to reach levels of severe impact that we observed in some area, I don't know, like especially in Apulia or on the almond industry, even at a lower extent in Spain, just to give an example, is relatively slow, which and this greatly, in my opinion, re-evaluates the importance of the role of the surveillance. Because in general, surveillance is considered something that, okay, it's something that has to be done, but when some pathogen arrive, it is very difficult to do something. But this observation of the long time required for the adaptation can re-evaluate the importance of this of the surveillance, in my opinion. Not only surveillance at the border for the importation, but I'm referring to of the surveillance on the territory, which, if well organized and with the proper tools, could give the concrete possibility to prevent the development in epidemics, maybe. I think it is quite important this, because now we are dealing with the severe impact of Xylella on the olive industry, in part also on almond industry. It is already something very bad. But there are other important sectors of the agriculture. I don't know, just the case of the grapevine industry, or the citrus industry, the fruit industry in general, besides the almond, that up to now are immune, are not interested by the attack of Xylella in Europe. And here there is another point, because at 8 years ago, there was a big fear in Europe for the risk to get of the what were considered frequent mutation of this bacterium, able to make the jump of species. Now, observing the fact that we discovered that Xylella has been introduced in Europe since already some decades, some tens of years, it means that also the important mutation, major mutation, able to make the jump of the species, can be very rare, or so. This again, in my opinion, should push us to focus more on improving the programs of surveillance. And in this, there is also we can find also a role of the research of the scientific community. We know that already during the project, concrete contributions has been done by the optimization of the diagnostic methods, or by improving the possibility, giving some technical tool that making now probably possible the early detection with airplane imaginary or something like that. But things that still, for this, there is, I think, still a room for improvement. And this improvement may be another point that could be afforded by the scientific community in the next future. Those are some considerations that we made.
If I can just add a few words to what you said, because of course I totally agree with you with the need to really emphasize the importance of the role of surveillance. And what was quite interesting in the talk we heard this afternoon is the very large range of symptoms we can have on olive trees, for example, with the very severe one due to ST53, they don't know strains, but the very least severe symptoms as consecutive to the inoculation of the multiplex strain that led to mostly dwarfism only, and not a lot of very important decline of the tree, but still it's not nothing. And Maria emphasized also the importance of the practices as for FS17, so resistant cultivar. Symptoms may develop quite quickly when the trees are not managed in a proper way. So it is very important also to ensure detection, proper surveillance of the plants, and also good management practices, of course, to be sure that the situation is properly managed. Maybe Pasquale want to add something. I know he wanted maybe to discuss a little bit about resistance.
Yes, sorry, I have written something because of the time. So we were asked to do not reinvent the wheel. That was the imperative that we were asked to follow during the last congresses. And indeed, we were asked to make treasure of the all available experience from our American colleagues. And I think we have followed this route from in XFACTORS. We have learned a lot in these two projects, prompted to XFACTORS, and we transferred the knowledge from one project to the other, and we got information from the bacterial side and also from the plant side. From the bacterial side, we have data about the outcome of infection in olives. So we know that besides ST53, also the Spanish isolate ST80 can be particularly virulent for this species. But I want to point also the interest also to the sandy strain CO33 that indicates us that other strains, not only Pauca, can be a threat for the olive in Europe. And something we are starting to know about the virulence determinants, and this was from what we learned from Ansi Card and Annalisa Gianpetruzzi work on ESVL. We need now to functionally test these findings. So we need to do mutants of Xylella to verify these findings. From the plant side, I think we have started to provide some answers to the diverse susceptibility of olive cultivars, as also you were saying before. Also to the mechanism of interactions. As from the Pierce disease, we observe the scale of susceptibility of olive cultivars, and the use of this information can be useful for the implementation of strategy from the practical point of view for new plantation, and also, as Maria reported, in the management of the resistant cultivars. And particularly, this is a very important subject because in Apulia, big new plantation of resistant cultivars has been made. As I anticipated, we think that resistance has multiple facets. There is not only one gene. We know that anatomical features play a role, as we discussed, of the size of the vessel, and but we think that the degradation process has a role also in the resistant, all the cultivars, and we need to go in depth in the composition of the cell wall, as has been done in other study. To me, just to mention some posters, but already I mentioned it, was very interesting. For example, a poster from Martinez Cabero, which was an approach to screen grape cultivars with markers for PDR1 resistant gene, the resistant gene from Gravan. Either from a practical point of view, and also to evaluate the susceptibility of local grapevine, because they are grapevine from the north of the Spain, to the susceptibility to this strain. And this argument, this subject, introduce us to a limitation of our work, a limitation that is becoming now very, very important. We did not have access, we did not have the genome of the cultivar Lecino. That is very important, and the availability of the genome of Lecino is very important, and we make, and I do, an appeal to the genetic community, which is an Italian genetic community, which is completing these studies, to make available this genome sequence, even if it is not complete. A piece of information, the one of genome of Lecino, which is very important now, also because to assist programs of classical and natural breeding, as you will see, for example, from the poster of Pier Federico Lanotte on the natural resistance found in the fields. And other piece of information, other piece that we need to investigate is to look for a better model system, because all of you have learned that olives takes at least 1 year to get symptoms, to show symptoms. And also, I was also impressed from I'm interested in I think it could be a good subject, the study of the roots, the xylella interaction in this multiplication in these organs, as you will see from another poster from our colleagues, Giuliana Loconsole. And finally, I am particularly happy that other players, which are not directly linked to the territory of olive growing, started to study Xylella in this species in Europe. These players, these colleagues, are proposing a transgenic approach. You will learn during the EFSA conference. And in perspective, I think that we should all pursue this route due to the observed limited resistance in olive that we have observed in very few varieties. I think that XFACTORS actively contributed to the start of the activities of these new groups that are new players in Europe, and also as well to the creation of a diffused Xylella scientific and practical knowledge in Europe.
Thank you, Pasquale. Maybe I can ask the speakers of this afternoon to participate to this conclusion if they have something to add or to comment. Maria, do you have something to add?
No, just to thank you that it was very interesting, all the questions that we posed and discussed, and the presentation. So.
Okay, I just have a look to see if there are some new questions.
Yes, there was another question from Joël Page. A practical question for Maria. So since Xylella seems to be detected all year round, should official survey campaigns also be conducted all year round instead of in summer only?
Yeah, this is a possibility that should be considered at least for those area with a mild winter, and for in the case of olives, that is an evergreen species. I think that in the upcoming days, Ofir Bahar from Israel will present similar results on almond, so supporting again the possibility to detect efficiently the bacterium in our case in olive, in vegetating plants, but in his case under the Israeli condition in dormant almond tree. So that could be a possibility which will enhance the phytosanitary authority to dilute the monitoring campaign, at least for some species. In our case, where olives is the main species monitored, this can be an opportunity to be considered.
I don't know what you think. Maybe if there is no more questions, and if we do not have anything to add, we still have 4 days to discuss and to ask questions if there are some. So maybe I leave the floor to you, Maria, to close this first day.
Yes, so just I want to thank you and Donato for chairing this session, and Pasquale for contributing to the conclusion on this important topic, which is the virulence of the strains, and on the other side the response of the plant. We are just in time, and we hope to continue in the next days with the same interest from the audience. I forgot at the beginning to say that the session will be all recorded, so those that were not able to follow the event today can find then the registration afterwards on the EFSA website and the XFACTORS webpage. I encourage you to see the proceeding, as we are not the book printed, but you can have a look at the proceeding that you can download from both websites, as well as I mentioned before, to contribute to the poster competition, and so to interact with the authors of the posters through Twitter and Zenodo. So I thank you all again, EFSA staff, and you, Maria Agnes and Donato, for this first session, and we see you all tomorrow at 2:30 p.m. with the session on vectors and the control of the bacterium. So see you all tomorrow. Bye-bye.